Okay, so we will start. Welcome everybody to this online conference organized by the PlasticHeal project and titled Shaping the Future of Micro and Nanoplastic Research, Connecting Science, Policy and Industry for Human Health. This is the second day of this conference, and as I commented yesterday, it is quite clear to us now that micro and nanoplastic pollution is a threat, represents a threat to human health and the environment. And it is our ambition to find out solutions so we can move towards a better environment and healthier lives. And for that, it is becoming more evident that we need to sit on the same table to the different actors involved in this issue, in this problem, to try to co-create solutions and to co-design what could be the better strategies to achieve this important goal, this ambitious goal. Out of the many strategies that we may apply to try to achieve this, one is to have better preventive policies and regulatory initiatives, and this is the role of PlasticHeal is really aligned with this. So it is our ambition to generate valuable information and valuable technologies that can be useful for human risk assessment, and then that can be applied in regulatory schemes so it's useful for regulatory purposes. So I'm going to make a very brief introduction of the PlasticHeal project for those of you that weren't connected yesterday. The project is following the rationale that you see here. We consider inhalation and ingestion of contaminated foods and air one of the main sources of human contamination, and we assume that these micro nanoplastics that goes inside the body reach the primary barriers, the respiratory and the gastrointestinal barrier, are able to translocate to the bloodstream, and then they bioaccumulate in the secondary organs where they can induce long-term effects. So following this rationale, what the PlasticHeal project is trying to do is to develop advanced systems related to each one of these categories, these steps in the journey of the plastic across the body. And then with this model, evaluate a bunch of different effects going from genotoxicity to gastrointestinal toxicity, respiratory toxicity, inflammatory effects, and large, etc. With the overall idea of later organize all this information into a framework that's valuable for regulatory purposes. So PlasticHeal is now ending in the last phase. The project will end at the end of March next year. So most of the models and most of the data have been already collected by the consortium, and we are ready now for applying all this information to a risk evaluation scheme and perform human risk evaluation. So we are ready to share this with you, and with the time that we have left, we would like to discuss two different phases on this, so we can maximize the impact of our developments. The first time that we will share this with you is in this conference. Yesterday we had a really nice discussion, and we expect the same for today. We will incorporate all the feedback that we receive from you, from the different stakeholders, the different actors, into our knowledge portfolio. And then we will discuss it again in the context of the cluster we belong to, with the cluster, with the other projects that have the same objective than us, which is understanding better the health impacts of micro nanoplastics. The collected input again will go to the consortium so we can wrap up and finalize the project with the maximum impact possible. So the conference has been organized in 2 days. Yesterday we discussed mostly about the information that we generated about the effects of micro nanoplastics and how we are collecting and organizing this information to evaluate the risk. And today the idea is to discuss more on the technologies that we have developed, evolving more around the exposure category. So I would like you to keep in mind that the aim of the whole discussion is to maximize the value of all these for human risk assessment and regulatory schemes. I'm going to very briefly summarize some of the key outcomes of yesterday's discussion, just for your consideration. I believe that you will have this available during the session, this information, so you can go and look to it. But just for you to know that we discussed about the framework that we have developed within the project, and several key issues arise, like should we consider exposure and how we should consider the exposure in the framework. We discuss about the potential of grouping micro nanoplastics so we can predict toxicity without having to assess all single types of micro nanoplastics that could be in the environment. We discuss about the possibility of assigning weights to the different categories of the framework, about transparency in applying the framework. There was a lot of discussion about the interest of having a threshold from the regulatory perspective. And there were also some comments in the chat regarding the need of standardization across monitoring, analysis, categorization, and toxicity from the regulatory perspective. We discussed also about the weight of evidence approach that we are using and how so we maybe need to fine-tune this. Discuss about potential uncertainty about the framework, how to assess the quality and relevance of the evidence that we are including in the framework. We discuss also about the role of artificial intelligence in the whole process, and several key knowledge gaps were raised, like the lack of representative materials, the lack of exposure information, and the need for epidemiological data. This is just the highlights of yesterday's session, and I wish today we have a fruitful discussion again about, in this case, the technologies and the exposure. And having said this, I will leave the floor to Virginia so she can introduce the agenda and how we're going to organize today's session.
Hello, good morning, and welcome back to those of you who were yesterday here, and welcome to those of you who are joining just today. I'm Virginia Mata. I am the communication leader of the PlasticHeal project. And well, the format of today's session is pretty much the same as it was yesterday. We will have several presentations followed by quick discussions, quick Q&A from you, 5 minutes or so for technical questions. And I would like to remind you that this session is being recorded, and the video will be shared on YouTube and social media of the PlasticHeal project. And feel free to participate on the discussions following each presentation. We have also set up a shared whiteboard that yesterday was the first time that we used it, and it will be available together with the recording and some of the materials that we are preparing for all of you to take a look at it after the meeting, after the webinar. Feel free to enter as well also the whiteboard to share your ideas, comments, or I don't know, share broader questions throughout the session. What else? Well, okay, yes, I would like to remind you as well that you might not see her, but we have a graphic recorder capturing the key points from today's session as well. And it will also be part of the material that we will be sharing with you. And as you can see from the agenda, what we will have today is a discussion about how to make the data available. And we will have 3 main presentations, starting with Alba's about how going beyond traditional approaches. Then we will have in vitro advanced models, how to go from in vitro advanced models to in vivo data by Hans Bouwmeester. And then we will have a short break if we want to. It will be decided along the way. And after that, we will have the final presentations by Thorsten and Irina Estrella Lopez from Helmholtz-Centre for Environmental Research at University of Leipzig. And we will start our roundtable, in which we would like to encourage you to actively participate as well, which will be moderated by Julia Catalán. And okay, so let's start with welcoming back again Alba. I was the pause between her presentation. So let's have her back again and start the meeting today.
Okay, so I'm going to start by putting this slide, which represents globally the different types of information that the PlasticHeal project would like to achieve. We are covering aspects about exposure, including external exposure and internal exposure in human beings, and we also are generating information on the kinetics and the dynamics of the behavior of these particles when they enter into the body. And we are generating also information about effects. So we are covering both exposure and effects in the project. Regarding effects, we are covering respiratory toxicity, gastrointestinal toxicity, some effects on secondary organs, the immune system, and long-term effects such as carcinogenesis. We are also trying to develop models so we understand better what is the relationship of these elements within exposure and also between exposure and the effects by modeling. So today you will listen from the speakers coming after me about this modeling, and you will listen a lot about the analytical techniques that we have developed and have applied to understand better the exposure. So I will focus on the rest of the things, so you have a global and overall idea of what are the different methodological approaches that we have taken in the project. So this is what I will summarize to you today in this presentation. I will start by talking about the test materials that we have focused in sizes below 1 micron, because we believe this is what is really important in terms of human health. And we have made an effort of going beyond the polystyrene beads that most of the publications are about. So we have tried to use other polymers and real-life materials, secondary plastic materials, nanomaterials. Then I will go to describe a little bit about the in vitro experimental models that we are using and about what we are doing with the effects perspective. Okay, so it was discussed yesterday, and this is something that it's always discussed in this type of forums, that we lack representative materials that really resemble what's in the environment. This can be a picture of what can be found in the environment, plastics of different shapes, different sizes, different polymer types, mixtures, etc. And this is what is mostly used in the lab, which are the polystyrene beads that are commercially available. So we've tried to move from here to here as much as possible to have information that is really linked to the type of materials that we are exposed to that can be found in the environment. So we have obtained nano PET from the degradation of transparent water bottles. We have optimized a protocol that was previously described in the literature, and by applying this protocol that consists basically in the degradation of the bottle and then applying a sequence of different chemical and collection phases, We have managed to obtain nano PET of around 200 nanometers, maybe a little less, but it tends to aggregate also quite a lot. We have managed to obtain it in sufficient quantities to really apply it or use it for hazard evaluation. We have also made an effort of characterizing very thoroughly the materials that we are generating. You have pictures here about the TEM and how these materials are really far from the bits of polystyrene that we are that most people are using. The shapes are irregular. And this makes a really completely different situation in terms of hazard. Also, we've managed to apply the same protocol or an optimization of the same protocol to obtain this time nano PET, but from the degradation of white milk bottles. These milk bottles contain an additive, which is titanium nanoparticles. So when we apply this process, we get in the end a particle that contains both plastics and titanium inside. And in these pictures, you can see how you see the titanium here, the plastic here. But even in those areas where you don't see the titanium, we know, as you see in this other picture on the right, that there's titanium within inside the material. So we are quite sure by the characterization of these materials, thorough characterization, that we are in our hands have a material that contains both PET and titanium at the same time. Another plastic that we have managed to obtain is nano PLA from TVAX. We have tried to follow a procedure that simulates more or less what can be the procedure of preparing a teacup. And by applying this protocol, we managed to obtain this nano PLA that you see here in the pictures with a size of around 150 nanometers. And all these three materials, this is just an example of the real-life materials that we have obtained, but we have used also others in the project, have been applied to understand the kinetics and the effects of micro nanoplastics in the different systems. It is something key for us to study kinetics. This means that we need to understand very well what's the internalization of these materials in the cells. We have analyzed this internalization, this interaction of the materials with the different systems that we have used in vitro. In all of them, we have done this in all our experimental work, so we understand better what is the effective dose, what is the amount of particles that really reach the cell. And it's also needed to study the translocation through the barriers. So we need to label the materials, so we are able to do that. And here in this picture, you have an example on why it's so important to understand or to study the internalization in all cases, because you see the same dose and the same material, when it's applied to the different cells, you see very different rates of internalization. In this cell, you see a lot of green, meaning that there's a lot of internalization, not that much in this other cell line, although it's the same material and the same dose. And here is the example of whole blood, where in the same system, in the same mixture, you have different cell types, and you can see very clearly how monocyte cells internalize a lot, and polymorphonuclear cells and lymphocytes are completely a different situation of internalization. That is why we give so much importance. If we don't control this at least thoroughly, we cannot interpret the results that we obtain with the analyzed effects. So that is why we also developed a method to be able to track the internalization, the translocation of these real-life materials. We use different dyes coming from the textile industry, And in the end, we realized that this one that you have here was the better in terms of not disturbing or compromising the cell's viability, and also being compatible with the other dyes that we typically use to analyze internalization. And this is an example of the application of this staining method with this dye to one of these particles, real-life particles, in this case the titanium, the PET doped with titanium. You see in green the plastic, and you see here in the middle that titanium piece that's embedded inside, and all inside the cells. So this is something that we value especially. Okay, so we have the test materials, we have the system to track the internalization, the translocation, the behavior, the interaction with the different systems, and what are the different systems that we have been applying in the PlasticHeal models. These are first related to the gastrointestinal tract. We have worked with transwell-based complex gastrointestinal models. You see here a scheme of what is the structure of these systems. These are grown in transwell. The cells are organized similar to the tissue, and if you apply different types of cells in here, you may have a mixture of that better resembles what's in reality. By applying this methodology, we have obtained barriers, such as the one that you can see here in the picture, that were really indeed very useful to track. Again, in green, the internalization of the particles that you can see here, and you can see here in these cross sections. One of the things about having these mixtures of cells is that each one of these cells comes from a different background. So the consortium has also attempted to obtain mixtures of cells from the same genetic background by doing these intestinal epithelial models from induced pluripotent stem cells. And you have here a picture of what we have obtained, at least 3 different types of cells from the very same initial donor. This downs the variability of the potential results, and we think that it's a better method for an attempt to move towards a more realistic gastrointestinal barrier. The same approach with the transwell has been also applied to the respiratory system now. Again, you have here the transwell with the cells that we grow here. The cells organize in a monolayer as in the tissue, they differentiate, and then we can apply the micro nanoplastic to study kinetics and effects. The good thing about the system that we have obtained here is that we have a window of more than a week, so we can expose these cells and try to observe what are the effects on the kinetics. I won't say on the long term, but at least at a longer time than 24 or 48 hours. You have here pictures on how it looks under the microscope, and it's really nice that we can have a clear mucus layer here, really recapitulating what happens in the pulmonary system. It is also quite interesting to compare here these 2 pictures. This is how the stratified epithelium looks like when we grow these cells in the transwell in an air-liquid interface, and look how different they look if we applied a normal system with liquid over the cells. So the way of organization of these cells of this epithelium is completely different if we change the conditions on one or the other side. So we believe this recapitulates better what's in the organs, and in the end, we can use it, as you see here again, to track the internalization and the translocation. You see a lot of mucus in this monolayer, but still some particles are able to translocate, to internalize and translocate through this mucus, through this barrier, to the basolateral membrane. The same that with the gastrointestinal model, with these respiratory models, we have also tried to move to another level of complexity by using organoids, in this case derived from human embryonic stem cells. You see here what's the result of this exercise that we have done, and how in these organoids we can also track for quite a long time, a week, even more, what is the behavior of the particles inside the organoid and what are the effects in these organoids. You see, in this case, in red, the labeled particles, and how we can assess here the exposure. This can even be more complex if we add functional macrophages from the same cell of origin, which is something that we have also tried. So we have organoids not only when one cellular or what the cellular types that are derived from the embryonic stem cells, but also the functional macrophages within them. So as a conclusion, I guess that we have tried to move from models of low complexity, simple cells, simple monolayers, to more advanced models recapitulating what can happen in vitro. And we have also attempted to include other factors that can modulate the particle behavior, the particle interaction with the system, and the particle toxicity, such as, for instance, digestion, the digestion process, the contact with pulmonary fluids, and the contact with foods, which impacts over the corona of these particles and changes completely the behavior. This is an example of how we have assessed the internalization of the particles being digested or not digested, and how the dynamics of internalization change quite clearly due to the corona that's formed in one or the other situation. Another system that we have used is what we call the ex vivo system of blood from donors. We use whole blood that we expose for, in this case, the only possibility is 24 hours of exposure of this whole blood to the different particles. And after that, again, we can use this system to assess for internalization, kinetics and dynamics to the system. We can separate the different cell types or white blood cells from this cell through flow cytometry, and we can then assess what's the level of internalization. This is an illustration, in this case, quantifying what I showed you before with pictures. And if you see here, for instance, monocytes in this ex vivo exposed blood, you see that the internalization levels of the different particles that you see here vary a lot, 41, 30, 70, 40, 85. So it goes from around 50 to around 85% of internalization in the same cell type to the different particles. So it is quite interesting that we can manage to understand this interaction better by applying these methodologies. Another system that we are using is the long-term in vitro system. I described this for you yesterday, but for those of you that are new today, what we do is to take a given cell type and expose these cell types continuously to a given micro nanoplastic. This exposure goes for weeks, even months. And the good thing is that we generate a cell collection of these exposed cells, because we freeze cells every 2 weeks. So we have in the lab a collection of cells that have been exposed to micro nanoplastic, and that can be used to analyze whatever endpoint we think is important. Not only a given endpoint, but we can also assess the mechanisms of toxicity. Let's say that if we observe that these cells have been transformed at this time point, we can go back and analyze what are the mechanisms that before the effect have been operated in the system. Okay, so after applying the materials to these in vitro experimental models, of course, we would like to understand the effects. And for that, we have tried to keep a balance of a combination of what is considered standard biomarkers of effect, like, for instance, genotoxicity. To the majority of these models, we have applied the comet acid or the micronucleic acid, for instance, to assess DNA damage of mutagenicity. But at the same time, we try to move to other types of potential effects, like the ones involving omics. We have analyzed the transcriptome, but also the proteome and the secretome and the microbiome, all the different systems that we have developed. And we are doing this because of two different reasons. The first is because we would like to identify relevant genomic pathways that are relevant for plastics, maybe specific for plastics. We would like to identify new biomarkers that are related to these mechanisms at maybe specifics for the plastics. But also we would like to gain acceptability of the models that we are developing. We know that it is not easy for the regulators to accept this type of systems that are not still standardized. So we are trying to make an effort of retrieving the mechanism of actions in a way of helping regulators to be more prone of accepting this model in the regulatory schemes. So for this is an example of the different omics data sets that have generated in the project, and those are, we have even more. So this is just a picture, a capture of some of them. We have a lot of different data sets that we can use to retrieve information regarding mechanisms and new biomarkers. We have developed a bioinformatic pipeline to be able to analyze easily all the information that we have, all these data sets together. And for instance, we can use this bioinformatic pipeline, for instance, to ask to all data sets what are the common gene pathways for a given micronanoplastic type. For instance, this is the output of asking this question to the pipeline about the commonly enriched pathways for PET treatments. This is real-life PET. So we have here all the pathways that are altered across the many different data sets that we have generated within the project. So it's a nice way of trying to find what are the specific genomic pathways operating in this toxicological profile. Another thing that we can do or ask to all these data sets is to try to look for new biomarkers with potential applicability in human biomonitoring. This is really important for us because in the biomonitoring studies, we are applying the standard biomarkers of genotoxicity, but we would like to see if we can do more. We can apply all the biomarkers that can also be informative of the effects that the exposed population is suffering. So we have a data set of single-cell RNA-seq of this donor blood that we are using. And we have the information or the altered transcriptome information of all the different subsets of cells that we have in this blood. So we can ask to the pipeline to retrieve, for instance, all the different genes that are commonly altered in the lymphocytes or the monocytes, which are the population of cells that is typically analyzed in the biomonitoring study. So when we do that, when we ask to the system to retrieve the different, the commonly altered genes in the white blood cells of the exposed whole blood, we have a list here of 8 biomarkers that we are now testing or trying to validate in the biomonitoring studies. So I hopefully have given you an idea of what is all this all the approaches that we are using that are not standardized regarding the test materials and the systems and the effects that we are analyzing. And we would like to discuss with you how we can do to make this information and make these developments useful for human risk assessment, because we believe we've done a lot of progress inside the project. We understand better what are the kinetics and the fate and the biodistribution of these particles inside the body. We understand better what is the hazardous potential of these micro nanoplastics regarding genotoxicity, respiratory and gastrointestinal toxicity, good dysbiosis, inflammasome activation, immune alteration, or carcinogenesis. We also have identified some potential mechanisms of action of these particles, and we have also optimized the analytical workflows to be able to retrieve human data regarding exposure and effects. We have also modeled, tried to model dose response and in vitro to in vivo extrapolation, and we have developed a risk evaluation framework able to organize all this information to categorize the risk. So all these achievements that we value a lot, we would like also that regulators value them as well. So this is what we would like to discuss with you. And with that, I would like to thank to the PlasticHeal team, also to the cluster, to the other projects inside the cluster that we collaborate together for this a similar aim. And thank you also all for your attention. And if you have some question, I'll be happy to answer. I'm going to stop sharing now.
And you're welcome to share your questions through the chat. Are you able to read it, Alba? We already have one.
Okay. There's a question regarding if we have faced different issues with low density micro nanoplastic samples. And also about the exposure of the Calu-3 system. Well, actually, I must say that we have not envisioned particular issues regarding the exposure. The particles, the monodispersed polystyrene or the real light particles that we are using behave quite well in dispersions. We apply the nanogenotope protocol for that. They tend to aggregate, of course, as happens with the nanomaterials. So we do observe when we apply these particles in culture that they sediment and they really reach the cells. So we have not faced particular issues regarding this. And regarding the air-liquid interface, what we do is to do a semi-liquid. We are not using a nebulizer. We just apply the minimum liquid possible, so we can still apply the particles in suspension. As you have seen in the pictures, we still have a really nice stratified epithelium. And regarding the question of whether our micro nanoplastic test materials are available for all the future projects, the answer is yes and no, meaning that it's really hard to produce these particles. And this was something discussed yesterday with the JRC that have offered reference materials in the past for nanomaterials. So it's really hard to obtain these secondary particles in huge amount in large amounts to be able to share it with large consortiums. So it is, of course, our intention to share as much as possible. We have established collaboration with many different projects and other researchers, and we are willing to share these particles, of course. But what I mean is that we cannot produce them in sufficient quantities to be a supplier, let's say, for everybody interested. So it has to be in petit comité, establish specific collaborations, I would say. There's another question talking about the long term, the long-term exposure, and if we see accumulation over time. So we have not really thoroughly explored the excretion rate, but we believe there's a turnover because it at a certain point, like for instance, after 2 weeks of exposure, the particles arrive to maximum situation exposure, let's say. So you don't observe, beyond that time, you do not observe that more particles more cells internalize the particles or that you have more particles inside the cells that are internalizing. I hope I have explained myself correctly. So there's accumulation during a certain time, and after that, we arrive to kind of a plateau. And we assume that this is because there's a turnover between internalization and excretion. But although we have not explored this in a deeper way. And then there's a question about the human data from the textile and the recycling industry, but I think that this you will receive a lot of information on this about the other members of the consortium that will talk after me. So they will explain how they have developed the methodologies to measure micro nanoplastics in humans, and they will give information about the results as well. I don't see any more questions in the chat, Virginia. I don't know if the whiteboard, have you shared the whiteboard through the chat?
No, but
maybe if we have some minutes now, you can do it.
If you joined yesterday, you'll see that it's a little bit more organized. Now you should be able to see it and interact with it.
Yes.
So you can use the first column to grab a sticky note and modify it as you like.
Okay, there's a comment in the chat. Some people may not have access, so please, Virginia, check that everybody can edit.
Okay. This will be the same whiteboard that we will be sharing during the whole meeting, so I will be applying the changes along the way. Please be patient. This is also a new tool for us. I will just leave it active for 2 more minutes. I'll try to see if I can activate edit permit for all of you. I see that some people are joining. Please, Alice and Hans, let me know if you can edit now.
No, still not, I'm afraid.
Okay. Censorship for you because you are a presenter. So
I think I can share my screen, but I cannot add anything to the whiteboard. I see that some people can. There's now a nice curve coming. Shall I start my presentation, Virginia, that you can perhaps in the meantime figure out?
Yeah.
I think it's a good idea.
Because I think there's also quite some overlap between the presentations, so that if people want to address anything on the whiteboard, well, either put it in the chat if I may propose, or save it for the whiteboard later.
Yes, I think that there will be enough time to answer questions, even though we will need to go back and forward to this discussion. But in the meantime, I think it is a good idea for you to start, and I will try to see what's happening with the whiteboard and get familiar with it in the back.
Okay. Let me see if I can share my screen. I think it works. Hopefully you can see my screen now.
I can see it.
Yes.
Good. So welcome. My name is Hans Bouwmeester. I'm working as a professor at Wageningen University in the Netherlands. I'm very pleased to be part of the PlasticHeal project and consortium. I have a little bit more a generic presentation, so not so much data, I think, as Alba showed you just a few minutes ago. Some overlap, but I think that's also good to reinforce some of the messages. And I will also come back to one of the questions in the chat on the, let me see, on the accumulation, because there are perhaps two approaches to study this. One would be, in my view at least, one would be experimentally. So can we have chronic exposure in vitro, long-term exposure in vitro, or could we use so-called pharmacokinetic models to extrapolate from in vitro results to human exposure, human concentrations, perhaps also effects in humans, using in vitro data, but then take this notion of accumulation, perhaps in the kinetics. But I will come back to that in one of my last slides. So let's move. Let me see. Yeah, there they go. I think it's highly relevant to, at this moment, discuss these particles. We all know, or hopefully we all know, that currently in South Korea, they're discussing the plastic treaty, and that's for a good reason. And perhaps one of the reasons is on this slide that shows all kind of the planetary boundaries. And one of them is, I think, highly relevant for us here in this meeting, and that's the transgression of the planetary boundaries by novel entities. And these novel entities are anthropogenic compounds, chemicals that could be pesticides are of course quite frequently being mentioned, but also plastic is frequently mentioned as one of these man-made entities that is brought into the planetary environments, and that it really suspicious, or I think we now more or less know that it is causing problems. So the larger plastics that we produce more and more, but also, of course, the resulting micro and nanoplastics. And an artist way, perhaps, to show this threat, if you want, is perhaps this. I found this nice monstrous cartoon on the internet. And you recognize perhaps the source of it. So there is a lot of plastic, of course, in our oceans, in our terrestrial environment, that is approaching not only the environment and appearing up from the environment, but it's also approaching us because we know that these large plastic fragments are degraded, and Alba already told you about this earlier this morning, into all kinds of smaller plastics. And you see them depicted here, just an assembly of that. And these are then still the larger plastics fragments, and you recognize the diversity in shapes and forms and colors. And what you do not see, but that's of course hidden in this picture, is the diverse composition in terms of polymer composition, additives, or other chemicals that might leach out of these materials. And that is, of course, highly relevant for us, and what we hope to discuss and have analyzed also, both with analytical approaches, but also with advanced toxicological approaches, Are there any interactions between these smaller or really the nano-sized particles that you can barely see also in this magnification on interactions with human biology? How do we want to study this? And the title of this, my contribution, is not so much to screen for all and introduce in all the different in vitro approaches, but move a little bit beyond that. So what do we have available in well-equipped toxicological or biology labs to study the potential hazards and interactions of these materials? And obviously, in toxicology, especially in regulatory toxicology, we would like to move away from using animal models. Sometimes it's very valuable because it's one intact organism where you can analyze both the uptake and processes that happen, as also was shown yesterday with the intestinal microbiome that might affect the uptake. But there are all sorts of reasons to also think and develop alternative methods, moving away from animal-based data, animal-based studies. And then, of course, it's a challenge to do these studies and study these interactions in humans. It's not ethical, of course, to expose humans to known dangers, to known chemical hazards. On the other hand, and it was already hinted to that, we are being exposed as humans, not only as consumer via our diet and drinking water, but also in an occupational setting. And that, of course, opens possibilities to screen in humans that are involuntarily being exposed to micro and nanoparticles to study the potential effects. Hopefully, still the exposure is rather low, and that might also pose challenges then to detect particles either in the lungs or in the intestinal tract or in urine or blood. So there is a good need for alternative models, and I would say human cell-based models, to really study the mode of action of particles on different cells, primary cells, cell lines, or perhaps, and I will come to that in a minute, and Alba already gave away a few of the approaches into perhaps mini organs where we have co-cultures of different cell types to emulate really the complexity of intestinal or liver or other organs in our body. Well, I guess you're all familiar with the 3R principle, and moving from animals to human cells can be captured, of course, in this 3R principle, where we want to the replacement or reduction and refinement, or the other way around, of animal testing in toxicology, in regulatory toxicology. What is now the approach to do this? Well, perhaps, and I'm overloading you perhaps with approaches and terminology, and I think this is one of the last theoretical slides, is what's called the adverse outcome pathway. So the toxicologist amongst us might recognize this, and this is a scheme basically that captures the biology leading to adverse effects. For instance, effects on liver, or you could also say effects on the immune system. If we now know how our liver functions and how chemical exposure or novel entity exposure could intervene with biological pathways that result in a misfunctioning liver, then we can start thinking, well, how can we now approach this also in vitro? What is the molecular interaction? Which molecules, how do particles or chemicals interact with a receptor on a cell? What is the cellular response? And if that goes out of control, how would that influence the different cells in an organ system, in an intestinal epithelium, in the liver, in any organ that's relevant for the adverse outcome? And that ultimately can then affect the organism, the health of the organism, or perhaps also at a population level. At the population level, we might not note it so early in human populations, but of course, if we would study effects on the environment, environmental toxicology, then of course population effects are also of large concern. So that's also depicted in these boxes at the bottom of this slide, but in a more simplified manner, I think also in the cells depicted in the upper panel. something happens to a cell, the cell responds, becomes activated, not only as a cell itself, but also in an organ where we have different cell types that work in a concerted action, either preventing or reinforcing the effect. If an organ is malfunctioning, you can predict that also at the individual level, effects can be noted. That is in a nutshell the adverse outcome pathway. And now the challenge is, and there are lots of debates, can we formulate adverse outcome pathways for small particles, for microplastics, for nanoparticles? And then it's often said, of course, that adverse outcome pathways are agnostic, chemically agnostic. So which are then the biological pathways that are relevant if you want to discuss the hazards or the risks upon microplastics exposure? And I think such a framework is helping us, is leading us in designing advanced in vitro models to study really using cells or mini organs, potential health risks to humans. Well, in a more simplified way to explain the adverse outcome pathway, perhaps you can look at the bottom of this slide and you might recognize the so-called Newton's cradles, where you move one ball out of the other balls, it hits the other, and then the effect is being transferred from one node to the other node, and in the end, you see that the ball jumps out, and that is then the adverse health effect on humans. Transgression, relation between the different key event and key event relationships, of course, need to be understood and studied. This adverse outcome pathway implies that we have models and responses that sometimes are simple and sometimes are complex. And obviously, the higher you go into this adverse outcome pathways, more organs or organ systems, the more complex you might want to have your in vitro models to study the effects. While if you are very early in the adverse outcome pathways, where you would like to study perhaps the primary interactions of particles or chemicals with a receptor or with a cell, you can use very simple models. And that's, I'll have a few examples on this chain from simple models. Not always we need to make it very difficult in biology or toxicology towards the more complex models. So how can we use simple models? I'll come to that in a minute. So which kind of models do we then have? And Alba already gave away some approaches. We can of course have simple models, a cell line. For the intestine, we frequently use, for instance, the K2 cell line. We can grow them in isolation or as an epithelium, or make it a little bit more complex, that we have multiple cell types to emulate the complexity of tissues from co-cultures, tree cultures, or exploit the power of biology. You all hopefully know that we in all our organs have stem cells. These stem cells renew our organs, renew our whole body, but we can now isolate these stem cells from tissues and maintain and grow them in our laboratory to either create organoids from lungs. Alba already showed them, from the intestine, and I will show you some intestinal examples in a minute. Adults-based stem cells or induced pluripotent-based stem cells. Both models have their pros and cons, but we can use both of them in our laboratories and study in depth the interactions of particles and human biology. But it's not only the biology that we can range from simple to complex nowadays. We can also use different technology for that. We can grow cells really simple at the bottom of a well plate, a standard plate. You can grow them in a trans well. That's the static cartoon. Perhaps you see my cursor. The static model, where we can separate different compartments, for instance, an outside or an inside, a lung, at the air-liquid interface, and obviously we can do similarly for the intestinal studies. But this is static. And in our body, we know that we have liquid flows and biomechanical forces that influence the differentiation of cells, and that is then entering the field of so-called organs on a chip, lung on a chip or gut on a chip. This has two main advantages. Also a disadvantage, but I will start with the advantages. The advantage is that perhaps the biology becomes more realistic. because there's liquid flow, shear forces are imposed on the cells, which might influence the differentiation of these cells to become more human-like, more tissue-like. And then there are different approaches, different technologies that one can use, and these are also depicted on the top panel of this slide. The disadvantage might be that it's getting more complex, more costly, so you can only do limited experiments, high-end experiments, if you really want to confirm perhaps effects that you see in simpler models, or that you see effects in animal models, but you really think, well, this is an interesting, highly relevant observation. Can we then confirm it using human cell models? It also opens the possibility, and I'm particularly myself a favorite of those approaches, where we can try to link different organ systems. And that's, for instance, in the commercial device, in the box circulation, where we can connect different tissues to each other, for instance, the intestinal tissue, and if we would make that leaky, perhaps upon the exposure to particles, then chemicals are taken up more readily in the intestinal compartment. What is then a consequence on, for instance, liver cells grown in another compartment? So now we can move up into this adverse outcome pathway also in our laboratory to study the potential interactions or disruption of tissues in one compartment and study the consequences on another compartment. Simple models. Let's start again simple. Early in the adverse outcome pathways, we are interested in simply sometimes screening materials. We have an awful lot of different plastics, different polymer types, with different additives, with different chemicals associated. We never can test them in a highly complex model, in the organ-on-a-chip model if you want. This is an example. We've done this in an earlier European project, and some of you might recognize it. The project was Marina, I think it was called. And there we used a very easy reporter assay, Calyx cell, which is a modified cell that upon activation of a receptor, it can be the estrogen receptor, it can be the androgen receptor or the HR receptor, which is known to respond to dioxins and PCBs. And in this example, we used a reporter cell line that is the NRF2 pathway, so that's a signal for oxidative stress. Oxidative stress, of course, has been frequently mentioned to be associated with exposure to particles. And this is an example where we use this assay, not for plastics, but we easily can do that, of course, but for different types of metal particles. And you see that we screened 3 types of particles here, and only the silver nanomaterials apparently induce the NRF2 pathway in a significant manner. And we've tested this simple cell model in different laboratories, sort of a pre-validation of this approach. So this could be also repeated, of course, with plastics. And I think it's a very easy way to screen different types of plastic, different kind of polymers, as I said, or different type of additives in these plastics to figure out, well, which combination perhaps is more hazardous than another one. And that's, of course, important if we want to move from simply understanding and observational studies to a domain, for instance, called the safe and sustainable by design. If we can then rapidly screen polymers and additives, then perhaps we can give suggestions to develop as well. Please do not touch this polymer additive combination, but focus more on another one. Simple models, quick answers, elegant to be used, in my view, for screening purposes. Then if you want to move up, and Alba showed you also a few pictures of this model, we could use multi-cell type models. For instance, for the intestine, and these are some pictures from my own lab, where we created monolayers of intestinal epithelium derived from iPSC, so induced pluripotent stem cells. And depending on the protocol that you use, you can create different combinations of different cell types. You all know, or otherwise I can tell you now that we have different cell types in the intestinal tissues. Of course, the intracytes, which are the cell types that take up the nutrients, potentially also toxins. Goblet cells in the middle of the slide, which are produced in mucus that is protecting the intestinal cells. Paneth cells are able to regulate, especially in the crypts, the cell division and the maturation of the intestinal epithelium. We have these three cell types now in our culture, but if there would be an interest for some reason to only have intracytes or to have more goblet cells or to also have other cell types that we know that are present in the intestinal tissue, there are protocols with different combination of small molecules that aid in the differentiation of different cell types. So there's a lot of variability possible, while we can also very nicely standardize these models and allow, well, it's more difficult than cell lines, but some reproducibility also amongst and over different labs. To show that this works and that it is sometimes relevant to use more complex models, you can have a look at the graph at the bottom of this slide, where we simply expose this complex model to, I think it was LPS, or you see the nice ricin and LPS, bacterial toxins related, and compare the responses in this complex model to a simple KCO2 model. And you see differences. Obviously, then the question starts, well, which model is then more predictive of the effects in humans? But I think there's also some value in having multi-cell type models. So this model is established, we can use it, and we did use it also for exposure to particles. Which particles to use? Alba already mentioned this also. Think of the novel entities, think of true-to-life particles, which are much more complex in shape, form, size than the routinely used spherical particles that you also see here in the middle, for instance, the polystyrene particles. It still is relevant to also look at these polystyrene particles because they have been studied so extensively. So you could also start to think of using these particles as a sort of representative material to see if we can use that to understand and, well, link responses in different tests in different laboratories as a sort of, well, validation. Of course, it's sometimes a loaded term, but to what's now benchmark responses in different cell models across different laboratories. But of course, we would like to see potential effects of the true-to-life particles. And this is what we've done. Here you see 3 endpoints that we analyzed in this iPSC-derived intestinal model. And 3 endpoints, the leaky gut, so that's the barrier properties. Oxidative stress has been measured in this model, or one marker of potential inflammation, the cytokine IL-8. We exposed these cell models to different particles. You recognize PS as the polystyrene particles, and you see that they do not cause so much effects. If you would have used positively charged polystyrene particles, then I would predict that we show dramatic effects. So that's also perhaps a good positive control to be considered. And the other particles are partly from PlasticHeal and partly from a Dutch project, Momentum. True-to-life particles, you recognize PP talc, PLA, PVC, the pentitanium that Alba nicely introduced earlier, and also polyamide. And then we see specific effects induced by some particles. So PP talc and PVC reduce the barrier properties, suggesting of a leaky gut. PP talc, the pentitanium, and the PA induce specifically oxidative stress. And again, the PP talc and the PVC and the PA induce the production of IL-8, suggesting of some inflammatory responses. But you also see that not all particles cause these toxicity. Polystyrene, and in our hands, PLA did not do so much. So that's, I think, important, given this my earlier remark on the safe and sustainable by design discussions. Some particles are more biologically active than other particles. It's always important to know the uptake. And Alba already showed you some nice pictures and direction, and we will come to that, I think, also in her presentation later in the symposium. So we can use in vitro models for the barriers, for the lung and intestine. We can use phagocytotic cells, but we need either fluorescent materials, and that's of course a difficult combination with true-to-life particles, or sensitive analytical methods. And I'm so proud of this consortium that we have developed, or colleagues have developed very sensitive techniques so that we now can move a little bit from only guessing what is the uptake to really measure the uptake in cells, also of particles that are not labeled. And hopefully my colleagues will dive into examples like confocal Raman microscopy, and not so much perhaps on pyrolysis, DCMS. So there are appearing now a panel of different methodologies that can be used here. But what do we need then to consider? Is it then only the difference between pristine particles and the true-to-life? Or do we also need to consider the interaction with the environment and the history of the environment that the particles have been in? And that is then opening a discussion towards what's also called the biomolecular corona. And what do we exactly mean with true-to-life particles? Are they then grinded from tea bags? Or should we also take into account weathered particles? So that's opening another discussion, I think, towards different complexity in the models, in the studies that we do. Oh, yeah, that's an example of models. So what does a cell see is then the question related to this biomolecular corona. And I hope that quite a few of you recognize that if you produce a particle, then it's pristine, naked, you could say. But as soon as you put it in a biological environment, it becomes coated, decorated by proteins. Proteins in your cell culture media, proteins from blood, if you would have biological availability, proteins from food in the gastrointestinal tract. And that we studied here. So we performed in vitro digestion experiments, simulating the digestive tract. We put particles in there. We confirmed that these particles with different types of biomolecular corona can be taken up by cells. And that's, you see in this confocal picture, the red is the cell membrane and the green dots represent particles. So we're really convinced that some particles are entering the cells, and then with flow cytometry, we can quantify the uptake and compare the different treatments. And just one example of polystyrene 200 nanometers at the bottom of this slide. The white bars are the undigested particles with the cell culture corona, you could say, and the darker bars are those particles that have first been in vitro digested and thus have a different biomolecular corona. And with particles treated as such, apparently in these cells have a much higher uptake rate than the others. So it does matter what the exposure and the history of the exposure is. And then of course we can analyze these proteins in this corona, and that you see in the middle panels in the A and B, comparing different types of plastics, the different types of proteins. We can analyze that at LCMS proteomics, and then try to correlate and understand, hopefully, the relation between the presence of a given protein and the biological effects. But that goes a little bit too far for this slide and this message, but it shows the importance, hopefully, of the history of incubation of particles. Another question could then be, well, is it then so important that we have different polymer types, or is the outside coating important? Or how is the outside coating influenced by the polymer properties of the particles? So I think quite some space to go in depth in these mechanistic studies. So all is fine. You can use different in vitro models, study the effects on intestinal cells, on liver cells, study the influence of the biomolecular corona on this, but this is all in vitro. And in the end, we would like to know, can we use these models, can we use this data to predict for safe levels in humans? And in chemicals, this question has been answered, well, I would say quite robustly, by the help of so-called PBK models. What is a PBK model? Well, you see that here in this slide, perhaps depicted. We have, of course, a human. You see the cartoon there, and we could transform this human into a computer model, into a PBK model, into the cartoon that you see here in the middle of this slide, where you have the different tissues connected to each other via blood, and sometimes interconnected. The gut, of course, feeds blood to the liver. And we can make these models and predict with such a model if we know the physiology of a human, what's the weight of a tissue, what's the blood flow to different tissues. If we understand some kinetic processes of chemicals, or in our case of particles, we can feed that all into this model and predict, for instance, concentrations of chemical, or hopefully soon of a particle, in these different compartments. So with a PBK model, we can predict the fate of chemicals, the fate of particles, concentrations of particles in different organs. Make a relation between the external exposure and the internal concentration. And in this graph on the right, you see that for chemicals, a PhD student of mine created PBK models for pesticides. And in the red curve, you see the concentration of a pesticide in blood. So you could similarly do this for heart, liver, for an organ included into such a PBK model. So that's nice. But can we now also do this for particles? And that is a little bit more a challenge, but I think we can. But with some more complexity and different equations, different biology or mathematical description of the biology behind these models. We know that there are cell types that accumulate particles in our body. These are the macrophages, the Kupffer cells in the liver, macrophages in the spleen, perhaps in the bone marrow. We know this. It has been shown also in vivo studies, rodent studies. And now the question is, can we then capture these processes, the biological processes, in such a particle PBK model? And I think that we can. Quite a few PBK models have been described that fit animal studies. But that's not too helpful, because then you use the animal data and use all that information and the kinetics there to fit a PBK model. But then you have a PBK model for a given study, while we would like to use a PBK model for different particles and integrate in vitro data for that. So we would need what's called a generic model. And I think we can develop that, and we are still working on that also in PlasticHeal in the few months that we still have in the project, based on a very nice paper that we found in the literature that used the physical processes of the movement of particles, that sediment on the wall of epithelial cells or endothelial cells, and all kinds of physical processes describing how particles behave in complex environments. And what those authors, and what we confirmed now, is that we really can use these generic models to predict concentrations in different compartments in our body, in plasma, in the brain, in lungs. You see a lot of different tissues where the concentrations have been predicted. The red dots are the animal data, and the lines are our predictions. And the beauty of this model is that we can run this now for different sizes. The model can deal with different sizes and predict the concentration of the different sizes. And if we advance these models, we can also take in more complex measures of particle exposure. So we are getting closer to a situation where we can run these pharmacokinetic models for particles. And that finally allows us to correlate the in vitro concentration response curves. As you see here, this is an in vitro concentration response curve. It could be in the intestine, it could be in the liver, it can be any tissue. If we then have a PBK model, then we can replace the concentration in our in vitro, in our liver cell test, to a concentration or a dose present in the air, or in the water, or in the food that we eat. So then we can really perform a risk assessment based on this. We can predict the in vivo dose response curve, and then set traditionally the point of departure could be an OEL or could be a benchmark dose level, to try to integrate in vitro data into a human risk assessment. This approach is getting more established and getting more accepted by, for instance, EFSA and other regulatory toxicology or risk assessment agencies. And I think it would be very interesting to push this one forward also for particles, to see if we can really integrate the in vitro data into this human risk assessment. And that, I think, yeah, that brings me to an end of looking a little bit ahead in the future, how we can use advanced in vitro models and combine that with these. I'll go back one slide with it, because it's, I think, such a nice summary, integrative approaches to correlate. And of course, we need to validate these models. And that's why it's so interesting to also have the human studies for occupational exposure, where we can predict, no, not predict, but benchmark, I should say, the concentration, the fate of particles in our body with these PPK models, with really epidemiological or occupational data. And I think I'll close it here, and I'm open for questions or suggestions or observations.
Thank you, Hans. You already have a couple of questions in the chat, and I will, in the meantime.
For me, Corona, I see. I have to read. Yeah, of course. So we can do simple digestions, and there are validated models published in Nature protocols. And I always have to think what was now InfoGest was the protocol, and there are also other protocols for that. And you can do that in a simple way, where you have the digested enzymes, but of course you can also include food or food stimulants in there to make it even more realistic. And that indeed can influence the composition of the protein corona. The challenge sometimes is, of course, to link this in vitro, because oddly enough, the bile acids in these in vitro digestion models are toxic to the in vitro cells. It's different from the in vivo exposure, obviously, otherwise we would not sit here. So there are also some practical limitations somewhere, but we can make the corona assessments more complicated. Effects of gastric fluids on the corona formation. And so, well, yes, I had one slide where we, I think, studied this potential leaching effects. Yeah, that's also interesting indeed, and that's of course heavily debated. You have the particles, true to life particles, that have either additives or perhaps chemicals from the environment adsorbed to these particles. Do these leach out? Well, we can do that in very stringent conditions to see under using stringent organic solvents as a worst case assessment, but we could indeed also perform these experiments in such an in vitro model, in vitro digestion model, and then study which chemicals leach out. And we've published work on that where we assessed this. Microbiome, yeah, interesting, difficult to study. Well, there are two potential effects, I think, either on the microorganisms themselves and and that's perhaps not, in my view, not the most interesting one, because there's quite some functional redundancy in the microbiome. But we can, of course, study the consequences of that. Do they produce different levels of metabolites, of butyrate or other important regulator short chain fatty acids? So that indeed can be studied. If you want to have a realistic in vitro model, then it's getting more complex from a technological point of view, because the small and large intestine, of course, is anaerobic. So then we would need a model where we can study the anaerobic growth of intestinal microorganisms while we have to provide oxygen to the epithelial cells. But there are models underway where this can be done, high-end models, interesting to explore further. I'm getting a error message for the whiteboard, I think. So let's see if I can read the chat. Then there's a remark on Fairytale project. I'm not familiar with that one. I cannot scroll down. There it goes. Lack of standardization. Yeah, that's always a difficult question. Sometimes, if I'm a little bit impolite, running the risk to be impolite, standardization perhaps is sometimes also used, not to go in the direction of more complex models. I know that there's standardization underway for the stem cell-based models, or at least to start with some sort of interlaboratory pre-validation. Also in the field of organs on a chip, people are very active in setting ISO standards. I'm not sure if that's the direction where Elisa, where you're pointing at. So also for these complex models, either on the biology side or on the technology side, standardization is underway. And then hopefully I've been answering questions in the chat. Feel free, of course, to put more. Or I think, Virginia, you've tried to make the whiteboard available. May I turn it back to you?
Actually, and we also have a couple of questions for the audience. Let me launch it.
Yeah, the data on the textile and recycling industry, I think, will come later, hopefully. I see that the question is coming up in the chat again. And we can indeed go to the questions in the poll. And of course, you should all prioritize in vitro studies.
There's also a comment about human data from textile and recycling industry. I don't know if you have seen it.
Yeah, but that's we studied that in PlasticHeal. Perhaps Alba, you can reflect on that.
Yeah, well, it will come with the next talk. I think we will cover these aspects because they will show how they have measured exposure in human data coming from the biomonitoring study. So let's leave it for the next speaker.
Okay, so the whiteboard will be still open, and you can just click on the link that I've shared in the chat and access it anytime. I will share the results of the first poll in a while. I will put it on the whiteboard as well. And in the meantime, we may continue with the presentations if that's okay with you. There was a coffee break programmed, but we can continue or do it now. What do you prefer? Or we can just keep the coffee break for before the roundtable session and continue with the presentations. I will leave it up to the presentation, to the presenters, to the speakers to choose whatever they want to do. The next presentation is will be by Thorsten Reemtsma and Irina Estrella Lopez, and I think they are both here.
And you ask us how to proceed?
Yeah, do you want to have a coffee break before the presentation?
We should have a 5-minute break for all the attendees who started at 9 already. Is that agreeable?
Amira is also wanting to take to grab a cup of coffee, so
yeah. So we should reconvene at, I don't know, 10:40. Is that good?
I think that people will appreciate that. Okay, so yeah. 10:40 we'll be back. I will leave. I will just leave the whiteboard open so that people can come in and play with that. So on.
All right. So good morning to all of you. My name is Thorsten Reemtsma from the Helmholtz-Centre for Environmental Research. And I'm also a member of the PlasticHeal Consortium, although I didn't manage to activate this background. And this presentation will be a shared presentation shared by me and Irene Estrella Lopez from the University of Leipzig, who will present the second part of this work. Yesterday we heard a lot, and today also we heard a lot and discussed a lot about hazard of microplastic, potential effects, models, and test systems that may be suitable to detect these effects. And this is, of course, a very important contribution that PlasticHeal has made to allow for improved risk assessment of micro and nanoplastic particles. But the second aspect that is essential for this is to consider also the exposure, to evaluate the exposure, to learn about the external as well as about the internal exposure to micro and nanoplastic particles, and ideally to do so by measurement, and to learn about the doses that we can expect. And at the end, of course, combining the two, the hazard and the exposure, is what we do in risk assessment. And this is where we want to learn on whether the doses that actually occur exceed the effect levels that have been established in the biological test systems. On the exposure side, the key challenges that we were facing in PlasticHeal is that existing data on the exposure to micro and nanoplastic particles of humans is very patchy and of variable quality. This was certainly the case when we started the project, and even today, when more data have been gathered all around the world, this is still the case. And based on this, we started our work on determining exposure to micro and nanoplastic particles. And concerning the external exposure that we first looked into, we decided to focus on oral exposure as well as on inhalative exposure, because it was considered that these would be the most important ways of exposure of humans to micro and nanoplastic particles. One specific challenge here in the external exposure is the high variability of this exposure of individuals, and literature is now also witnessing this. You see here a collection of data on the indoor and outdoor air exposure, which varies by orders of magnitude depending on whatsoever condition. And the same is true even for drinking water data. This was only recently compiled, and showing us that also with the drinking water, the exposure may vary by orders of magnitude. So it's not an easy task to determine the external exposure of humans. And our key questions here, we're focusing on two aspects. Our practical studies, we're focusing on two aspects. One is, what is the potential external exposure via drinks, which were stored in plastic bottles? This is, of course, a common assumption that this could significantly contribute to oral exposure of humans to micro nanoplastic particles. And the second aspect that we are working on in PlasticHeal is about exposure at workplace, occupational exposure in comparison to exposure of the general population. And I will show you a few data on this and hurdles and achievements that we have made in PlasticHeal with relation to these two aspects. Concerning the internal exposure, then, of course, there's also a variety of data that has been collected in the last years. I show you a graph here from a very recent study compiling such data and giving microplastic concentrations per gram of tissue or milliliter of blood. And there was a very recent study on microplastic in blood that seems to confirm the data given in this review paper. And if we look at this and calculate from the data given in that study, this would mean that 10 to 30 milligrams of microplastic would circulate in our body. And you may either find this alarming or questionable. So this was the situation for the internal exposure. Also here is strong patchiness, quality discussions, uncertainty about the validity of data, and the variability of exposure between individuals and between different groups of persons. So our key questions here were, what is the internal exposure actually on one side of occupationally exposed, probably highly exposed people, individuals in plastic industry, and also of the general population, which is not occupationally exposed. Much of the unclarity we still have today about the exposure, the external one as well as the internal one, is due to analytical challenges that we face when we want to quantify micro and nanoplastic particles. We have a wide variety of sizes, from micro to nanometers, that we looked at in our project. You may even think of millimeters, but this is probably less relevant for human exposure. We have a wide range of different polymer types with potentially different effect to humans. We have very variable sample matrices. In our case, the drinks, blood, other human tissues, air samples, and so on. And we luckily, at least in the case of internal exposure, face very low concentrations. Sometimes we may even have to detect single particles in the subsample that we have to process. Another challenge comes from the size issues, because micro and nanoplastic particles are well the size of many biogenic particles, of cells, of whole organisms even. They may even be embedded into biological tissue. And if we look at size, this is the size range we were focusing on in our project. So it's from below 1 millimeter to below 1 micrometer, actually. So it's a orders of magnitude size difference that is also, of course, analytically challenging to cover. Based on this, we took a lot of effort in optimizing sampling processing strategies that are able to release micro and nanoplastic particles from the samples they are embedded in, which are able to remove all the disturbing matrix in those samples, whether it was drinks or blood, for example, remove disturbing matrix, and in the same case, in the same time, preserve the few microplastic or nanoplastic particles that are found in those samples. That was a lot of effort, of course, and we then also decided to use complementary detection methods to cover the whole size range that we were interested in for the micro and nanoplastic particles. And I will show you this complementary approach on the next slide. Now, here you see the different approaches for cleanup, which were enzymatic cleanup as well as chemical cleanup, filtration on anode disk filters, and then basically the project relied on spectroscopic methods to detect single particles and to identify them and to count them, actually, the single particles, either by infrared spectroscopy for the larger particles or by Raman microscopy for the smaller ones. We used other methods for backup, which are mentioned here, but most of the work was done by these two methods I just mentioned. And in this way, we had a uniform sample processing approach shown on the right-hand side at the top, and then separated the samples, and those went to the micro or to the nanoplastic analysis. So it's really a combined and complementary approach in quantifying these particles in the different samples that we were processing in the project. Literature tells us that the outcome of such exercises of quantifying micro and nanoplastic particles much depend on the methods that are used and have been used in the different studies. And so we also attended an interlab comparison test that was organized by this cluster, actually. And you see here some of the outcome, which is submitted now for publication. And indeed, data can be highly variable. It to some extent depends on the methods that are used. The red method seems to have a higher variability here, for example, in these data, than the green one or the blue one. It also depends on the labs and the processing, of course, of the samples. But this interlab comparison also showed that there was indeed a comparatively broad number of labs that actually produce data in what we would call here the green range, which seems to be highly agreeable and comparable. And we were lucky to be in the group of labs that met this green range. So we had good results. In total, these were 80-something labs contributing to this test system, and which gave us confidence also on in the validity of the data, of our data compared to data of other labs. And now let's move forward and look into results that we have gathered. And this is the workflow we established for the oral exposure via the different drinks. We basically focused on 3 kinds of drink: milk, so cow milk, and tiger nut milk, and also orange juice. And even there we needed different, you may think, oh, all these is drinks and basically they are based on water, so what is the issue here? But we need to develop different cleanup and processing procedures for these 3 materials, because the composition, sometimes it's more with fibers, sometimes it's more with proteins and fats, and we need to adjust the cleanup for each of these matrices for a good recovery and for the ability to later on detect the micro nanoplastic particles from these drinks. The outcome is shown in brief on the right-hand side. Actually, for most of the drinks, we did not find this significantly higher concentration of in this case, microplastic particles in the drinks, not for the milk and not for the tiger nut, compared to the drinks stored in glass bottles, at least. While for the orange juice, maybe a more aggressive fluid after long storage periods, it seems to be that the microplastic particles concentration was a bit higher. This was all on polypropylene, because this was the bottle material in which these samples were stored. This was a controlled exercise and experiment done by our partners in the project, which we then analyzed. A large effort of this consortium, PlasticHeal, was also to learn about the inhalative exposure, and this was done in 2 respects. One respect, occupational exposure in industry working with plastic, and this study here covered 2 non-woven fabric manufacturers in Spain as well as in Finland, and also 2 plastic recycling companies, again, one in Spain and one in Finland, so in different countries with different processes being performed. And we compared the exposure there with urban exposure, short-time measurements in France and long-time measurements in Finland, and also with samples collected in a rural or even forest environment in Finland. Altogether, this was more than 90 samples which had to be processed, and given the complex requirements in sample processing and measurement, this was indeed a large effort for the consortium to process and analyze these samples. And I can show you a bit of the outcome here. It's a highly condensed overview of the data, which basically tells us on the external exposure in these sites that the exposure in these processing industry, non-woven fabrics, as well as plastic recycling, was in the range of 500 to 600 or 700 particles per cubic meter on average. However, if we go to the graphs in the middle, we see these column graph. The first column here, we see that this exposure was not equal. The microplastic and nanoplastic particles were not equally distributed in the air, of course, but at different sites in these manufacturing companies, we could record different concentrations, lower in some places and much higher in certain other places, so that maximum concentrations at certain processing steps reach several thousands of particles per cubic meter even. Concerning the identity, which is the second heat map kind of graph here on this slide, concerning the polymer identity, the 2 fabric manufacturers showed highest concentrations for PET, which is indeed the fiber that is produced there. and lower concentrations of other particle polymer types, while in the bottom, the two factories doing plastic recycling, we see a more diverse pattern of exposure with polystyrene and polypropylene in the one case and polyamide more prevalent in the other case of the factories. And this is certainly what you would expect for plastic recycling, that you have a more diverse exposure of also of the humans via air in these workplaces. Size-wise, and this is now the third column, the median range of sizes was between 20 and 40 micrometers, quite uniform for the different size, not absolutely uniform, but comparatively uniform, with little frequency of larger particles above 100 micrometers in size. And these levels of exposure that we found in these occupational exposure that we found here is about 1 order of magnitude higher than the exposure in the urban environment, and about 2 orders of magnitude higher than in the clean air, the forestry environment samples that we processed in this project. And based on these data, we also did an aggregated exposure assessment, telling us that indeed the occupation on the data that we have collected in this PlasticHeal consortium, not data from literature, telling us indeed that the occupation, in the case of the occupational exposure, the inhalative uptake is the dominant, probably the dominant exposure pathway, while for the general population, which lacks these occupational exposure, also oral uptake may be of relevance for the total exposure. And inhalation, of course, was highest there in the occupational situation, compared with up to 80, more than 80% of the exposure potentially by inhalation compared to the other routes of exposure for the occupational workers. And I will stop here and now hand over to Irina, who will continue with the work on the nanoplastics done in PlasticHeal. And I first stop sharing. Thank you for your attention, and we discuss probably together at the end of this presentation. Irina, are you there?
Yes, yes. Just a second. So now I'm going to share. So are you able to see?
Yes.
Okay, perfect. Good morning. I'm going to present the second part of this presentation, and I'm going to focus on nano-sized particles. So there's concern about plastic particulate matter, especially about toxicologically relevant micro nanoparticles, smaller like 5 micrometer. Particles are mainly getting into organism by inhalation and gastrointestinal routes. They can interact with the cell, overcome biological barrier, and translocate to secondary organ. And of course, we can expect the response. And particle can degrade, decompose, release of contaminants. They have nanostructure, means they have very high surface volume ratio, can interfere with different intracellular biochemical processes. From other side, they are able to act as a vector, facilitated the entering of organic and inorganic pollutants. That's why it's very important to understand model of action and to perform a reliable risk assessment. So we need valuable information about exposure that can be useful for human risk assessment. That's why there's need to develop and validate reliable, valuable, novel screening tools for detection and identification of plastic particles, especially nanosite fraction of them. I give a small overview about different analytical techniques. It's not all techniques what I'm presenting, but it's more important and more reliable and state of the art. So important things, so many techniques are able to chemically identify polymers. So it is molecular spectroscopy like Raman infrared or attenuated total reflection Fourier transform infrared. Molecular imaging consisted of micro Raman, micro FTIR, micro ATR, FTIR, and another techniques, we can call it like thermoanalytics, is pyrolysis, gas chromatography, mass spectroscopy, or thermal extraction and desorption, GCMS, or difference scanning calorimetry. So you see that all of them are able to identify the chemical identity. Some of them able to follow additives, get information about surface of particles degradation. But more important output, actually, this is the last three rows. The particle number, concentration, size, shape, and morphology, which is very relevant to risk assessment, it is only able to be made by molecular imaging. So, and in this case, thermoanalytic failed. The limit of size detection or spatial resolution, you see this is very different. So we have by Raman, we are able theoretically to detect the particle up to 250, not up, highest 250 nanometer. By FTIR, we are able to detect only a size larger as 20 micrometer. Of course, you can go to synchrotron radiation FTIR. It's very expensive, time consuming, and you can go down to 2 micrometer. This is possible. So, and very important information, mass concentration as well. So you see that molecular imaging can recalculate it. And thermoanalytic is able to give this information about mass concentration of particle, but not able to distinguish whether it's just polymer or it's this particulate matter. So, and I'm going to focus on molecular imaging, Raman-based molecular imaging, and to try to detect nanoparticulate matter. So this is a small overview what I'm going to speak about. So I will show how we have developed and adapted confocal Raman microscopy for characterization of micro nanoparticles in complex matrices. I will focus on the region between 50 nanometer and 100 micrometer. So, and I will show the application for internal human exposure, for biomonitoring. So biological fluids, blood, sputum, exhaled breath condensate, urine. We have analyzed different population in different environmental and occupational setting. Also, we look at the worst case scenario for the patient with a chronic kidney disease before they started dialysis. Another group is external human exposure, so food, water, occupational exposure, air and recycling, and textile companies, and of course compare it with the environmental exposure in urban or in forest. We are also involved in internal exposure in vitro in ex vivo. It's work package 3 and 4, but I will not go in detail about it. This is the people that were involved in my group in this project. So very shortly, what does it mean? Confocal Raman microscopy is coupling between confocal microscopy and spectrometer, and in each pixel we are able to detect fingerprint of specific biomolecules and that allow us to show the distribution of biomolecules, like here DNA, lipid, cytochrome, and so on. We are able also to detect that plastic in ex vivo. It was in cooperation with Manchester University. Cyril has reported on it yesterday. And of course, we were able to detect not only biomolecules, we were able to detect particulate matter in complex matrices like milk or human fluids. So, first of all, we have tested sensitivity of confocal Raman microscopy towards single particle detection and limit of size detection. So as you see, we were able to detect, identify, and visualize single particle range from micro to nano down to the 50 nanometers. So we were able to overcome an optical resolution for nanosized particle by using pixel single detection and intensity-based visualization. The second, we have developed different analytical strategies to detect the particles. First of all, we have used enrichment by filtration. If needed, Thorsten has talked about it, about digestion if you need it. And at the end, we have bright field images and a molecular image, and we're using particle scouting algorithm to identify each particle and chemically identify and to get information about shape, size, and impurity, and so on. Another approach was optical trapping enrichment. So we have used laser tweezer effect to be able to detect single particles in the laser focus. It's called a flow Raman detection mode. Then we have tried to validate our method. So as you see, particle you see particle mass on membrane as a function of applied particle mass. So you see slope 0.97. So it means you have recovery around 97%. So we have a just experimental setup, particle density on filter. We have used different approach for calculation of concentration, improvement of image processing procedure, and at the end, we've got a limit of detection, let's say for filtrated volume 10 milliliter, 15 ppb, and it's pretty good. So the next approach that as I told, it's a flow detection mode. So we are able to detect single particles, and you see up to 0.3 microgram per milliliter. The particles were able flow constantly in and out of laser spot, and in this saturation region, you see the particles just accumulated here, and it's just impossible to quantify anything. So the calculated limit of detection was about 10 ppb. So then we have validated Raman microscopy for milk. Thorsten had spoke about digestion procedure. We have digested and then filtrated our spiked milk, and you see the distribution of polystyrene, and here's overlapping image of protein with polystyrene particles. So you see the rest of protein actually don't really disturb us. So we are able to distinguish these two signals and to look only on polystyrene, or in that case, on polystyrene particles. So the recovery was about 87%, and limit of detection was similar to the water. We have validated for the blood, sputum and urine, and we have looked also on plastic distribution of a blood component and blood smear. So the particles were mainly internalized and localized with immune cells. Could also found in vicinity of erythrocyte and serum. Okay, the next slide, I'm going to show an application of confocal Raman microscopy for human biomonitoring. And first, I will look at the external human exposure. The samples of orange juice and milk in plastic bottle and in glass bottle we have got from Barcelona, University of Barcelona. The idea was to analyze an impact of food design, treatment, type of packaging, or storage condition on release of micro nanoplastic. So how does it look? You see this bright field image, and you can find many particles. But if you look at the red points, this is points that was somehow related to the plastic, because everything else is something different. So we have combined this bright field imaging and screening with Raman, with the chemical imaging, Raman imaging. So you see here polypropylene, titanium. So at the end, summary, so we have found that in heat-treated milk, so it was like simulation of sterilization of milk at 120 degrees for 20 minutes. So it's very roughly a description. The plastic bottle consists of polypropylene and titanium, and bottle lid consists of colored polypropylene. So in milk, in heat-treated milk, we have found 300 particles per milliliter of polypropylene. We have found also titanium, PE, and pigments. And in orange juice, the concentration was more or less 10 times higher. Oh, yeah, exactly, yeah. For polypropylene, and the fraction of nano-sized particle was about 30%. So in the glass bottles, we didn't see too much particles. It was really very low concentration of particles. So next example of next example of application, it was external human exposure. We have analyzed air and textile and fiber company in Alicante. And as expected, we have found a lot amount of PET and titanium oxide particles in the air, in the fabric. A large fraction, more like 50% of particle had nano-sized. You see the quantification and here size distribution of PET and titanium on aspect ratio. Both particles are formed like fibers rather than spheres, because the aspect ratio was 0.5. Then we have analyzed the work of it on the same company and the same company in Suominen, and it just to show you how does it look. This example from one worker. So we were able to quantify the composition of different particles. You see PET, 31%, titanium oxide, different kind of organic and inorganic particles. You see the size distribution, aspect ratio, and quantification of particles. The same we have made in whole blood samples. So you see high concentration of particle has been detected, up to 2,000 particles per milliliter. The range of particle was between 500 nanometer and 4 micrometer. Both were found in the breath condensate and in blood, and the fraction of nano-sized particle was up to 30%. So if you compare it with with the blood from patient with chronic kidney disease before they are starting dialysis, you see completely different plastic pattern. So you have huge amount of polyester, but I wouldn't like to say that it's very preliminary data. Nevertheless, what was alarming, we have found many Teflon particles, what is not good. The last slide summarized the preliminary results of human biomonitoring and showed particle abundance in occupational and environmental setting. So We have analyzed 3 fabrics. So the first fabric was in Alicante, textile and fiber company. So what you are able to see that in the air and in the human blood, we have found PET and titanium oxide. Furthermore, that polystyrene and polypropylene have been found in breath condensate and in whole blood. So, and the recycling, as expected, we have found polypropylene in the air, and also we have found it in the blood. And then the last fabric, and it was also textile and fiber company in Nakila. We have tried to see impact of environmental on occupational on the human fluid. So we have compared the particles that were collected in air by Finnish Occupational Health Institute. And it was forest not far away from Nakila, and in Tampere, urban area. It's not far away from Nakila as well. So what we have found, that environmental setting has higher impact on plastic composition in human fluids than occupational setting, unfortunately. So you see, and strong overlapping of polypropylene, PE, cellulose, pigment, and titanium, and you can find it also in the human samples and in the air. And the last bad news, titanium oxide and Teflon are omnipresent. We have found it in all samples. A very short overview about concentration. So we are speaking about up to 5,000 particles per milliliter in the blood, in the range between 300 nanometer and 5 micrometer. And if you compare it with literature or what Thorsten has reported, normally the people have found between 2 and 3 microparticles plus minus 5 per milliliter. And the average size average is 130 micrometer. So just to compare it. Okay, now I'm going to the conclusion. The 2 molecular imaging tools, so micro FTIR and confocal Raman microscopy, were adapted and developed towards chemical identification, visualization, quantification, and characterization of micro nanorange plastic particles in environmental and human matrices. Both tools have been optimized and validated for detection and quantification in complex matrices, and this is more important output, the recovery around between 80 and 100%, and we have limit of detection. In case of FTIR, it's 4 particles, and for limit of detection, in our case, we have 1 particles, and or limit of detection and quantification in PBB, it was around 15. So the both tools are complementary and have been successfully applied for chemical identification and size shape analysis or size shape related screening of plastic in occupational and environmental setting, to quantify internal and external human exposure, because it's paramount important parameter for risk assessment. So, and of course, we need more data on human exposure for risk assessment. In case of FTIR, there are many attempts to harmonize and standardize FTIR, but in case of confocal Raman microscopy, we need harmonization and standardization of Raman microscopy towards identification and quantification of nanofraction of plastic particles, because they are toxicologically relevant. And of course, we need of interlaboratory trial, but joint approach using different state-of-the-art tools in regard to their applicability for plastic monitoring in complex matrices. This is what would be good to do, and it's probably very important issue for the future. So thank you very much for your attention.
Thank you, Irina, and thank you, Thorsten. We will now open the 5-minute space for technical questions as well. You can use the chat, and I will share again the whiteboard, and also some questions for all of you.
Mm.
So as we are late anyway, maybe we then proceed if there is not too much to ask right now.
I have just one question. Virginia, you was not able to see my slides, but other people were able.
Certainly, yes.
Okay.
We will share the slides after the
Yeah.
meeting, and I apologize because we're supposed to be sharing everything with everybody, and I'm not sure what the problem is in terms of permissions with the teams. We have a comment from Alice. Are you able to read it, Irina, Thorsten?
No, I don't see it.
It's on the chat.
Yeah, I'm in the chat. Wait a minute.
It was on the Interlab. It's related to the Interlab.
Okay.
Certainly,
from Alice,
certainly it's not possible to answer this in general, whether other labs have improved their methods. I can't tell you.
Now I am able to read the question from Alice. So the question is, I was interested to see you mention an interlaboratory comparison. Okay, do you think there has been improvement in the methodology since this project? Yes, I think so. I mean, we've got money from you. We should improve the method. Of course, we have improved a lot. I should be honest, I didn't believe at the beginning that we will be able to identify and quantify any particles in the blood. And due to improvement and different approaches, we were able really to go down to the limit of detection and quantification, and really to be able to quantify a low amount of particles in spike samples, as well as real samples from internal and external exposure. Yeah, now I'm reading the question from Tina. Hi, Tina. I didn't see you a long time. So, is there a chance to further improvement to detect even smaller nanoplastic? So even smaller. This is a question about time. What you need to detect it. So I have shown that we are able to detect 50 nanometers. So, but 50 nanometers, of course, is under optical resolution. That's why you cannot use fast approach by using particle scouting. It means that you should visualize it first. You should visualize particles optically visualized. So it means you're already immediately limited, and if you have bad contrast, probably to between 300 and 500 nanometer. And this is a limitation. But if you going to molecular imaging, you will be able to detect the particles smaller particles. But anyway, you cannot say is it particles have size 50 nanometer or 250, because you see the optical pixel and you see these very small particles, but you have no information about real size. But you know, of course, it could be developed. We have started to do it intensity-based analysis. So you have calibrate your device and know what intensity you would expect from one single particles, which says size 50 nanometer. But it is really very tricky already. But it could be done, and it's very time-consuming.
Okay, you can still, all of you, continue commenting on the chat. And I want to thank you, Irina and Thorsten, for the presentation. And as we are really late in our schedule, I would like to welcome Julia Catalán, who will be moderating our roundtable. Maybe after we still have time to, I don't know, make another break. That's up to you, Julia. And I don't know, let us know what you want to do in this case, and then we can just pause for 5 minutes or continue with the roundtable.
I will suggest that, well, hello to everyone. I'm Julia Catalán from the University of Zaragoza and also a member of the PlasticHeal team. And I will try to moderate the second roundtable, as Virginia mentioned. So maybe, Virginia, if you could show us the composition of the second roundtable, I can already introduce the speakers. We do a really brief break so they can be ready for questions, and then we will continue. So we have today 4 speakers in this roundtable. Penny Nymark from Karolinska Institute. Penny is a toxicologist, and she has a lot of experience about omic data and adverse outcome pathway and how we integrate them in the regulatory context. So I hope she will tell us about that. Bjoern Hidding from the company BASF. This company, as all of you know, is manufacturing a lot of chemicals, nanomaterials, and also polymers. And Bjoern, concretely, he has experience on regulatory background in hazard and risk assessment. So we will have, which is very valuable, the point of view of the companies that is also provided by our third speaker, Amira Fernández from Implast. Implast is also a partner in PlasticHeal, and she has experience in risk and efficacy assessment of polymer materials. And she has really, she is really working in the lab. So she has experience what are the problems when doing in vitro bioassays. And our last member of the roundtable is Alberto Katsumiti from Geiger Technological Center. Alberto is working in one of our sister projects in CASP, Plastics Fate. He is also, he has background in environmental contamination and toxicology, and he has also worked a lot with different types of NAMs. So now I will suggest that we have 5 minutes break, and then we will come back and start the roundtable discussion. So 5 minutes will mean up to Virginia.
4 minutes, and we can round it to 35.
Okay, 4 minutes. Take a quick coffee.
Okay.
Right. Okay. So if you don't mind, my idea is to address, let's say, one question to every of you based on your expertise, but of course, all of you are welcome also to comment about it. And for the rest of the audience, if you want, your contribution is also welcome, but what you can do is to put in the chat any question you also want to address, and then we will open also the discussion broader. Okay, so Penny, I will start with you. Hello again.
Hi.
And as I said, you have ample experience with omic data and especially also with the adverse outcome pathways. Yesterday, when our framework was presented, I think it was Blanca who pointed out that several of our endpoints are usually mainly based on in vivo data, and it's true. Carcinogenicity, reprotox. So my question to you is, in the current status of the adverse outcome pathway-orientated assessment, how much or which degree of evidence do you think we could reach with the NAMs that have been presented today?
Okay, that's a very complex question.
I know.
And I had planned to begin from a very general aspect, but of course, as you say, I have long experience with omics and experience with AOPs since they became a thing. So, and everything I'm going to say builds on that. So that's my starting point. But what I want to start maybe with saying before I directly answer your question is that it's all about trust and using data for regulation, but also for safe and sustainable by design is very strongly linked to trust. And NAM's new approach methodologies, which of course then are, for example, omics, and which link to AOPs, or the hope is that by linking to AOPs, you increase trust. These methods are still in a stage of disconnection from the regulatory endpoints, as you said. And this is a challenge, naturally. I don't have a direct answer to it. I do see aspects, because it all builds around trust, I see aspects as validation, not necessarily full regulatory validation, but a type of scientific confidence as an important aspect. But I also see something else that is more and more relevant today because of, and we see it or we understand it perhaps better because of AI, and the AI increasing and sort of becoming the next step. You know, bioinformatics that was used combined with omics originally was seen as some sort of magic bullet. We were going to answer all the questions with this amazing informatics approach. Today we have AI, and we tend to talk about it in the same way. It's going to be a magic bullet. It's going to give us all the answers, but it won't do it unless we give it good data. And so I want to stress all the amazing data that you have, and I want to thank every speaker from yesterday and today. Really amazing project and a lot of amazing data coming out of this. And I would urge you to take care that this data becomes fair. And when I say fair, I mean findable, accessible, interoperable, and reusable. Many of you have probably heard about this acronym. But I want to maybe stress the fact that fairness does not necessarily mean that it's going to be fully applicable to regulatory approaches or to safe and sustainable by design, which of course is more targeted to the industry. But it's going to make it possible for them to assess whether it's useful. And the more we move towards understanding what quality means in the process of safe and sustainable by design, or in the process of regulatory risk assessment, the more we can do these assessments if the data is available and reusable in good ways. So, and not to mention interoperable. So all of this, going back to omics and AOPs, all of this builds on the fact that omics is fairly fair. So omics is one of the few data types that are stored in very standardized ways and connected to fairly good metadata, but it's not connected to all types of data out there. It's mainly described for itself within its own domain. And AOPs naturally also have a need to allow for integration of data. So all of what I've said connects very much to these two areas. But I urge the level of fairness to maybe be considered within the project.
Yeah, thanks a lot. The suggestions, yes. What about the other speakers? Do you have any comment to my question about, I mean, especially industry? We are really interested on your point of view, how you handle this. You have to, in your case, you are working with polymers, you have to provide information, you are a regulatory link. Can you manage with NAMs? We are not yet in this stage. What is your opinion about, please? Feel free to tell us.
Yeah, maybe I can start. I would very much agree what Penny just said. I mean, I saw a lot of great data today and yesterday here, and thanks to all the researchers. And I think if we put all these bits and pieces together, we can start to form a broader picture. But if I would put myself into the shoes of a regulator, I would ask, how urgent is it to regulate, and what is it I should regulate? Should I regulate at a chemical level, which would, for example, then be a polymer reach, or is it a product level where I say, hey, let's take the low-hanging fruits like single-use plastics, like we had it in the EU. So this is a little bit what we should consider when we think about regulation. And to be honest, I'm quite unemotional here in which direction it should go. If it is really a chemical regulation, then we would of course need some kind of intelligent testing strategy. And I think we could make a lot of use by in vitro methodologies. We have seen today a lot of good modeling approaches like PBPK, IWIFE, which is also used in the chemical space, so non-polymeric already. And the EU Commission is setting up a NAMS roadmap at the moment, and I think we can learn a lot from it, how to use this data, and maybe then we can also come up with a robust idea how to use this data for polymers too. But it really depends on the politicians, how they want to approach the regulation. If it's not a chemical regulation, then we might need less data compared to to a product or article related regulation.
Okay, thanks a lot, Björn. Amira, do you have any? Sorry, Alberto, I think Amira was first raising her hand.
Hello, Julia. Hello, everyone. No, in fact, it was something about they were talking already. It's true that now I have one question just to also introduce myself to everyone, because I think maybe some people I met last May here in IMPLAS. I'm Amira Fernández. As Julia was saying, I work in the in vitro bioassays laboratory, and I'm very grateful to be here, learning a lot about all of you. And it's true that IMPLAS, as being technological center, we are very close to the industry and very close to the regulatory, in my case for food contact materials, but also thanks to my colleague Juan Fran in the regulatory of microplastics, or at least trying to standardize the methodology to evaluate risk assessment. But now I have some questions also to debate everyone, and also to have feedback from everyone, because it's a chance for me also to be here with you. Chemical regulation, but when we talk about micro and nanoplastics, we can include them in chemical regulation, because sometimes it's also challenging the pretreatment of these micro and nanoplastics. In polymers, also for food contact materials, is something that is happening the same in the pretreatment materials, and it's something that we are working hard in the intercomparison also with different labs to try to get the reproducible data. So my question also now here from a technological center that maybe has industries that can ask this question. Micro and nanoplastics can be included in chemical regulation. I don't know if it's
this is a question I cannot answer, but obviously, but maybe we have in the public even some people linked to regulatory issues, or I don't know if Penny wants to answer this, but I don't know how it is. I think it's considered as pollutant.
Yes,
but of course, on the other hand, it's true that for the industries could be very interesting if they could have some clues and thinking about safe by design, how some products or how some polymers could be designed in a way that the pollution is reduced. So in that sense, I think that independently, what is the official consideration, I think we should consider like within chemical safety, because everything starts from the beginning. What we produce is what we find at the end.
Yeah, I can say very quickly, just regulation is often performed in the way that it asks for information requirements regarding what is going to be put on the market. And so if you don't know if you don't have a picture of the life cycle of the product, then it's very hard to know what to regulate. And I agree with this question on what to regulate, and that's where safe and sustainable by design comes in. But of course, and which is not a regulation, it's probably not going to be in the sense that we see it, at least in line with the framework recommended from the EU or the Commission. But at least it's a way to understand better, but it's very difficult to get it harmonized and to get everyone to engage without regulation. So we're a little bit in a chicken or egg situation.
No, yes, it's true. Thank you, Julia and Penny. Also because I was remembering one of the first slides from Alba at the beginning of the day of today, that there was this design that connects everything: research, citizens, consumers, industry, regulations. So also it comes up how education or how the dissemination is important also to enforce the needing of this regulation.
Definitely, yes. Alberto, I think you have been for a long time with your hand raised.
No problem. Yeah.
Sorry.
So welcome to the discussion.
Thank you very much. And I think just to start, I would like to really acknowledge, to say thank you for the invitation, Julia and Alba. It's a pleasure to be here, and as a new event, for example, for the collaboration between the different projects, for example, the CASP is a pleasure to be here. It's a pleasure to discuss these different questions, for example, here, and represent, the Plastics Fates project in this discussion. So I think the question from Amira is very nice and very good. And actually, I think it's very nice to see, for example, all the discussion concerning the SSPD, and the plastics as well. Sometimes the discussion on SSPD and plastics are very separated, but I think they should be discussed like in the same context, of course, because when we think about plastics, we have to think that we don't have only the polymers, but we also have the additives in the plastics. We can design the plastics, we can design the different polymers, for example, for the different applications, and choose, for example, these different cocktails, for example, for the applicability, but also for the safety and sustainability aspects, to design in a way that we can keep like the functionality, for example, but then having, like a more safe or a safer and more sustainable, plastic at the end. But I think, focusing, for example, in the main question, the first one that we discussed, so for example, is basically like the NAMs, or the use of NAMs. So personally, for example, we are bigger supporter on the use of NAMs, for the hazard assessment, for example. So in Geikia, we work with NAMs for ecotoxicity. We work with NAMs for human health as well. and a bigger supporter, because I think we can use these NAMs at least like in a pre-phase of the regulatory, approval of these different products that can be very, very useful. But I think it's important to raise another question, which is the data quality and also the use of certain NAMs for the hazard assessment of plastics. I think along these 2 days, we saw very nice and very good data. Some of this data, they are not completely new for us because we saw in many presentations from PlasticHeal, that this data is really well done and that the collection of the data and the experiments are really well done. But we know from the experience, not only working with plastics, microplastics and nanoplastics, but also with nanomaterials, that setting up experiments with these particles are not very easy. So there are several challenges that need to be taken into account before the collection of the data. So we know that some experiments, they were really well organized and really well performed. But depending on how you set up your experimental system, so maybe you can reach some data that is not really accurate, or more, less or more accurate. So I think some of the questions, for example, especially in the chat, raised some of these aspects that I know that, for example, in PlasticHeal, you guys are doing really well. But for example, the assessments of endotoxins on the particles is a key thing, so not everybody is really doing this, and sometimes you can see some studies link, the increase of inflammation, for example, with the presence of the exposure to microplastics and nanoplastics, and we know that some materials are highly contaminated with endotoxins, so maybe these effects are not really related with the plastics, but with this parallel contamination, for example, dispersion of the materials is very key as well. We can have materials that can float in the in vitro system, and then they are not really accessible to the cells. So dosimetries, I think, is a key thing, and I think so. These aspects should be taken into account before knowing that these experiments can be used, or these NAMs can be used. So I think it's very, very important to really ensure that your NAM or your experimental method is really applicable, the applicability is really correct to assess this, and then ensure that the data quality that you're getting at the end of this. So, everything that we normally discuss, for example, especially, for example, in the nano world, if we can use a certain method to assess the toxicity, for example, of nanomaterials, this discussion needs to be happening here as well. So long story short, I think really believe on NAMs, but we really need to be very careful and very critical on the applicability of these NAMs, and then ensure, to ensure that the data quality is really good, and at the end.
Yeah, thanks a lot, Alberto. Somehow you have already answered some of the questions I was thinking to address, but thanks a lot for that. Penny, short comment before I change to the second question, please.
Yeah, I thought that was a really great statement by Alberto. And I just wanted to point to the benefit of safe and sustainable by design and the approach in terms of applying NAMs. So I want to just give a few examples of how I would see it as a researcher who has been long involved, but who has also a little bit of regulatory experience today. So the benefit of SSBD is that it's fairly, or a little bit more flexible towards the application of NAMs, because it's not dependent on the information requirements that come from regulation. And so it is, of course, it costs, so there's always a hesitance to use it, especially if you're not sure about the NAMs data quality. But there are, so it's still dependent on scientific confidence, in contrast to perhaps regulatory risk assessment, which is more dependent on legal certainty, which is a lot more robust, or requires robuster approaches. But there are a lot of, for example, when you think about omics, there are OECD guidelines for how to report omics data, or how to report around the omics data and the testing method that was used in order to generate the omics data and also the analytics around it. And once you use or implement those a little bit more, it becomes more robust also for use within SSBD. And this goes also for example, the EFSA qualification system for use of NAMs within food and feed and also pesticide risk assessment. And I think these are all jointly approaches that will support us to raise the confidence in NAMs and NAMs data. And connected to that, I also want to bring up something that within regulatory risk assessment is quite thought to be one of the most successful alternative approaches, which is grouping and read across. And this is naturally also highly applicable early on in an innovation process during the SSBD, because it allows you to use data that is not necessarily fully predictive of a regulatory endpoint, but it indicates similarity. And omics is one of these areas that even regulatory guidelines are starting to look into, to using it not for predicting the whole uncertainty of predicting an adverse outcome, but to use it as a type of bioactivity-based similarity assessment, which gives you not full confidence in a risk assessment, but it does give you guidance and support for decision making during an innovation approach. So, you know, we kind of need to just push towards using these things on different levels and slowly increasing the confidence, and perhaps that also then goes a bit slower, but feeds into the regulatory approaches and change of regulation, which is slow.
Okay. Yeah, it connects, thanks Penny, because it connects with, let's say, my second question that, well, I will originally address to Bjoern, but of course, again, all of you can answer. Just to make sure all of you participate. I see there is not any problem, but I didn't know in advance, sorry. So, okay, so going on with using NAMs, Bas has a great experience also with for nanomaterials. So can we use the same NAMs that are used for chemicals and for nanomaterials in connection with the comments also that Alberto raised? Do you think that there is something specific for plastics, polymers, that we should take care and maybe some NAMs are not applicable? Because of course the use of validated NAMs, if we could use something that is already validated and is accepted for the plastics, then we could speed up a lot.
Yeah, that's a very good question. And I think Alberto already phrased it quite well. And you mentioned the validation. I think it's important to know for what purpose you could use a whatever methodology, and to have a little bit of feeling on the chemical space, but also on the borders and boundaries of a methodology. And we have both experience in nanomaterials, but also in NAMs here. And it really depends on the method and the substance or polymer you want to test. I once had a test with a nanomaterial for reach purpose in aquatic environments, and we had 2 years of work to establish a constant exposure in the aquatic environment. And this was hilarious work. So you always have to ask, what is the answer that or the hypothesis that I want to prove, and then check if the test systems available can offer you this answer for your chemical. As I said before, it's still some way to go, because I think a robust validation of NAMs is needed. What I could imagine to do also in a regulatory environment a little bit is some kind of a sandboxing approach, that you take certain assays, etc., and just test them without any regulatory implication. And then you can also go to risk assessment, etc. This is quite a new approach, but I think it makes use, because then all stakeholders come together, can share their experiences, and can come to an agreed outcome, which will also make it easier for regulators to really set up something meaningful in maybe less time than we have now. As Penny mentioned it quite well, regulation takes time, and if we can give confidence from all stakeholders also to the regulators, then they might come up with more informed decisions in shorter time periods.
Okay, thanks a lot. The other speakers, do you want to comment something on this? Alberto?
So I think initially just, I'm so sorry, Julia, for putting so many topics on the same time, like answering the question. But well, I think it's really well connected, and probably the whole discussion, like in the roundtable, will be connected with this different aspects, right? Because they are aspects that we deal, for example, when we work with microplastics and nanoplastics as well. So I think it's really well, explored the points that we discussed here right now. And for example, the applicability of the norms is basically this. And I think it's as you mentioned, so it's really to understand the boundaries of the applicability of these norms. There are plenty of methods, that we can use, for example, depending on the nature of your contaminants or your particles, right? So we really need just to know, like, for example, which are the methods that we can apply to certain types of materials, nanoparticles or plastic particles, and so on. And what are the methods that maybe are not really the appropriate ones to use with these particles. In some cases, for example, we know that some things can be applicable, for example, for microplastics, and for the nanoplastics is more like a challenge in some cases. But I think really understanding what are the methods appropriate to do this assessment is good. I would like to just really stress the fact that I think preparation of the materials and ensuring that everything is really well done in terms of performance is very, very important as well. Alba mentioned, for example, in the beginning of her presentation, the use of the nanogenotox protocol, for example, to suspend the particles in the case of the in vitro experiments. This is really well done. We know that, for example, even in plastics fate, we use the same protocol and works quite well for plastics, which is very, very good. Then maybe my question could be, for example, to the members of the roundtable as well. So what could be like a good method to use, for example, for the ecotox sites, in terms of exposure? Because the fact that we use BSA, for example, to suspend these materials and to stabilize these materials, these plastic particles, is of course linked with the composition of the blood. So this is very human related, right? But what should we do for the environmental side? Should we use the same approach, or should we adapt another, environmentally more relevant method, for example, to suspend these particles? It's very experimental, but maybe some of you have some ideas.
Yes, please, Amira. You are mute, Amira.
Sorry, thank you, Julia. Thank you, Alberto, for your explanation. I was also thinking in nanotalks, and also, for example, here in INPLAS, in our laboratory, we work with the guideline from the EFSA published in 2021 for nanomaterials. So we follow this workflow from the characterization, identification, also seeing how the particles are stabilized and do not agglomerate in biological fluids before treating the cells or before doing the in vitro digestion. And the battery of bioassays that we normally do here are comet assay and micronucleus, as Irina showed in the presentation also. And I think this workflow that the guideline from the EFSA about nanomaterials is very close to the harmonization of the techniques, because we are also reducing the use of animals in vivo assays, because if we conclude that we have negative results of the genotoxicity, also oxidative stress is also negative from our nanoparticles, we can conclude and we can not use animals. Then about your question in ecotoxicity, it's very important because from all the information that we can get from PlasticHeal, we can also build a PlasticHeal 2.0 in the environment, because as you were saying, I don't remember the person in the presentation that was saying about the additives and all the components that can be added in these micro nanoparticles that can then be released into environment, and this is very important for the contaminants that has to be analyzed also for this safe and sustainable by design in the reuse of these materials from the environment, from the compost, from the biodegradation. And here we are working now with a bibliofishery, okay, and also with fish shell lines and with primary producers, okay, from the say of fruits plants, and then with the tertiary consumer that could be with cacochu and FG2, so we can cover more or less the trophic chain. So this is what we are trying to do now, also because the future of the strategies in Europe over the reduce of animals in experiments to be more sustainable, also with these techniques, is also to go to a zero pollution strategy in air, water, and soil. So for 2050, the regulation also will be very clear with all these three scenarios that we can find in environment. So also we have to focus moreover the human health in environment, because when environment is affected, we are also affected, because it's something that is going to be included in our food, in our soil, in the products that we are going to eat. And we have to be clear also for the industry, also for the consumers, and also for the society that maybe is not close to all these topics, that maybe the danger comes from all these components that are linked to the micro and nanoplastics that then can be released in the environment.
Yeah, thanks a lot, Amira. And yeah. Okay, yeah, this raises another issue that listened today and yesterday to the speakers came to my mind. Yes, yesterday we were commenting about the I think it's a new guideline about the water quality and the assessment of microplastics in water. And Thorsten has also mentioned about the interlaboratory assessment that has been done at the gas level. But there we are talking more about microplastics. I think that the limit in water, someone pointed out that is like 20 microns. But then when we are coming to relevance for human health, in many cases, for instance, inhalation, probably only particles below 5 microns, as Irina pointed out, are relevant from the exposure. So how can we solve this problem of the exposure assessment for human? Because I think that there we have like a dichotomy between what is the progress in the environmental level and what is the progress in the human-related level, in addition of this different composition of the media that you have pointed out. So what can we do in addition of validating methods, but if we are not still there, how could we assess human exposure? Because if we don't have human exposure, we cannot do risk assessment, obviously. So what is in general your opinion? This is an open question to all of you and also to the public, if there is any good idea about. So someone brave enough to answer?
I actually don't know much about this. For the water directive that is on its way, I have not been involved in that, and I'm not sure how thresholds are considered there. But at least human exposure and setting of thresholds and safety limits, it has always been a kind of some level of acceptance of risk, even if we are so now that we are in a situation where it's already spread, it's out there, the safety limits need to be set at some feasible level. And this is what's happening for PFAS, for example, which I'm sure many of you know. And we can never clean it out of the water, or we can maybe, but at the current state, it's very difficult to get it away. So the limits have to be feasible, which in the end is the question, what do we do in this case? Because we know we should have precautionary principles in place, and we should avoid as much as possible spreading. But it's very difficult since it's so much, the sources are difficult and all of that, and the whole safe and sustainable by design is still questioned whether it works and whether it can be put in place. So I don't have an answer. I was just braining out. I think it's a very difficult situation. Although I do have a feeling that we tend to forget the past sometimes. And I know many people say this often. We should remember there has been problems of particulate matter in air before. We've put regulation and measures in place to limit that to the extent possible. And the same goes for this. It's complex, but we can't assess all the different types of materials that or particles that are getting out there. So go the other way around. And I think we heard a little bit on that previously by many speakers, what to regulate and how to regulate, and have polluters pay or all of that. So yeah, no answers, mostly speculations.
Okay, thanks, Penny. Björn?
Yes, well, I would fully agree to what Penny said, and we have heard a lot of approaches who could at least tackle this issue from one or the other side. You can try to model the internal exposure, and I mean we have different exposures here. You can check how is the bioavailability, where would it end up, etc. So you can come from that side, and then you have this biomonitoring part where Thorsten presented up on. And I think maybe if you take some really tangible cases like the workers' exposure, the occupational exposure in a recycling enterprise or something like that, maybe then you can find a certain common ground. But in the end, if you want to have a threshold level, you would also need the hazard data. So if you then take the inhalative exposure, you somehow need a toxicological value where you can say, okay, with an assessment factor of this and that, I can end up here. And my suggestion would be, why not getting in contact with some of these bodies or experts who set these threshold levels to ask them for their advice? Because, I mean, we have seen there is certain exposure, we have seen certain effects. So ask them, here you have the data, what would you still need to derive a threshold level, and maybe start at the occupational level, because we have seen that the exposure is quite high there. And then we can derive some experience how to tackle this issue. But as Penny said, it's very, very complex. You need to have exposure data, hazard data, you need to have enough confidence. But yeah, I would really go and ask the experts, like the former Skoll board or like that.
Okay, thanks a lot. Amira, some comment on that?
Yes, no, just also as my colleagues talking a little bit about the situation. it's also important when we talk about the exposure of plastics in general, that we are talking about the material with the most restrictive regulation, because it's everywhere in construction, in food contact materials, in health. So it's important also to remember that from the regulation and policy, they are all working very hard in the regulation of the new problems that we are seeing in the modern life. But it's also important to remember this, that we are talking about the regulations that are the most restrictive also for the clients and the industry of plastics. So we have also to be to have hope in this consideration. And also talking about safe and sustainable by design, it's important that as consumers and as society, sometimes the exposure comes from a not good buy of the things that are in the market. I mean, we all know all the problems that can come from brands, and I'm not going to give any name, okay? But in the internet, you can find all these delivery systems that can deliver food contact materials that they have not passed any regulation. And the problem is that the consumers are buying many products from all of this. Also with the textile is going to start all this problem, and the textile is very far away from a regulation, from a correct regulation. So the problem is not the material, is that we have the work now from universities, technological centers to work together to request this regulation to use safe materials.
Okay. Any brief comment before, because you have your hand?
Yeah, and I wanted to ask, I saw some comments on omics in the chat, and I was just wondering if you want, I can perhaps give a brief overview.
Yes,
Is that okay? Or
yes.
it jumps a little bit maybe in the discussion?
No,
and of course, because I also just briefly, I also see, the question is how much NAMs and test methods do we actually need in terms of nanoplastics? Is it maybe more an aspect of seeing whether we can come up with some precautionary approach in the end? That's the question. But when it comes to omics, at least, I do feel, so there was some questions here in the chat regarding the predictivity of omics and how that, usually it's classically been a snapshot in time. It's often related to maybe one or a couple of doses, and it's been more of a mode of action type of analysis rather than anything that relates to risk assessment, which needs dose responses and points of departure and all of that. And I just want to say that that's the aspect of predictivity of omics in terms of endpoints, regulatorily relevant endpoints, is not the only thing that it can do. So it's not only about the prediction of what might happen after time, but it can also function as I said, for defining points of departure, and not only also as a basis for bioactivity similarity, which doesn't indicate any kind of prediction, but rather is a snapshot of what happens in a particular system when that system sees particle or chemical or anything, and that is used merely to relate to well-studied reference materials or chemicals. So it's more of there's a broader range of what you can do with omics today, and I think that's the mechanistic part of it is a benefit, but it's not the only thing that you can do. So I just wanted to clarify that. I won't go on. I have a lot more, of course, to say regarding omics,
sure.
but
Yeah,
I won't
thanks a
use
lot.
all the time on that.
Thanks a lot, Penny. And again, you brought up an important question. We were also yesterday talking about reference, or better said, representative materials. I think a common problem we have with nanomaterials is that we don't have any positive control on many of the assays. So now I will address the question first to Alberto, who has been silent for a while. So based on previous project, and I'm telling that because we were working together on that, do you think that how could we, for NAMs, for in vitro model, we were trying to establish some methods between with nanomaterials, with some representative materials that could represent like the highest dose and that we could use to define thresholds in our systems. Could we do something similar with the micro and nanoplastic? Do you think that, because everyone was yesterday asking about the thresholds, the systems, but how can we define the thresholds?
Yeah, so I think here we have to remember, for example, that, I mean, we still lack standard reference materials for plastics, right? So this is something that is still like a complicated thing, because, for example, for nanomaterials, I think we have some options of representative materials. So I think the ones that we use, the most, for example, for many projects are the representative materials from the JRC, which are good options, and that we have many groups working with these materials. We know that they are very good for some things. They are not ideal for other things, as we know, and we assess, for example, in many projects, for example, presence of endotoxins, for example, in some of the materials coming from the representative, the materials from the JRC. So in some materials, we can see that the materials, they have low levels or even like higher levels of endotoxins. So they are not completely ideal for experiments. They are definitely good as a reference, and they are, absolutely great, for as representative materials. But in the case of microplastics and nanoplastics, we still lack, right, standard materials to have as controls. Not only saying, like, for example, positive controls necessarily, but something to compare with your material. So some benchmark, for example. So most of us, when we started to work with microplastics and nanoplastics, we start with the beads, so using different sizes. But of course, these are very artificial plastic particles that can behave completely different in the cell culture medium, and can, generate completely different responses as well, because the shape is rounded, it's perfect. So not necessarily very similar to what happened in nature. So I think this is still a big lack. And this is one of the things that we are discussing quite a lot, for example, in the project, so in Plastics Fates, but also in the different meetings in CASP, right? So the need of standard materials, the need of representative materials for these micro nanoplastic experiments. We still don't have it, and we need.
But this is just almost a question also for everyone. Could we consider that the micro and nanoplastic are close enough to nanomaterials that we could use the representative nanomaterials for somehow establishing this threshold in our test methods? I don't know if you understand my idea. I mean, for nanomaterials, we are using chemicals as positive controls because we don't have any other thing. So the question is, could we use nanomaterials, what we know as Alberto has defined, could we use them at least to help us to settle some kind of threshold in the different assays? Since we don't have any plastic focus representative material?
Can I start maybe? Well,
yeah.
Penny, you
No, go ahead.
Faster than me. If you want to go first, up to you.
No, you go.
All right. So I think in my opinion, I think it's very complicated to use, for example, nanomaterial as this benchmark, because I think it's important to remember, for example, in the case of microplastics and nanoplastics, coming from plastics, we have a very complex mixture, right? So we have the mixture, we have the polymer, but you also have all the additives in this microplastics and nanoplastics. So we have to think about, for example, that is not an inert necessarily, particle, so you can have the release of some chemicals from these particles. We have similar experiences, for example, with nanomaterials. We can have some particles, for example, that can dissolve, releasing ions, but it's not completely the same in the case of plastics. So we know that, for example, some of the additives, they can migrate from the microplastics and nanoplastics and then induce some effects. And this effect is not a single molecule, but it's a cocktail of molecules that is very complicated to really relate, well, in plastics phase, but also in parallel projects. For example, we have this approach. We're exploring this. So not only the exposure to microplastics and nanoplastics, but also from the exposures to the additives. And we can see that it's very complicated to link the effects that we see, for example, in the cells or in the organisms with the particle per se, because we have a mixture of different compounds. And these different compounds can induce, for example, endocrine disruption or neurotoxicity or inflammation that comes from this mixture of these additives. So in my opinion, it's not really ideal to have this. What we used to do, for example, especially, for example, in plastics phase, is to have the classical negative controls and the positive controls that are chemicals, for example. And then we can maybe use a reference for particles, but they are not really close to the microplastics and nanoplastics. They are just a reference in terms of shape and size or something like this for internalization studies, for example, but not really, as a proxy of these microplastics and nanoplastics. This is our experience in this project.
Okay, thanks. Penny?
Oh, I saw Amira having her hand, but you're okay.
Oh, but you were first, so go ahead and then I go.
Well, thanks. So I wanted to point. I don't know. I don't know enough about nanoplastics to be able to say whether it's going to be good to use the materials that we have within nano safety. But I do want to point to the fact that the JRC representative materials, it's good to remember that they represent the first generation of nanomaterials that we started with 20 years ago or something. And they don't necessarily represent the current state of advanced materials that are very quickly being developed. And I wonder, in that sense, whether nanoplastics isn't part of, it's not necessarily an advanced material because it's not purposefully generated as such. But if nanoplastics shouldn't be part of that endeavor to figure out whether the current OECD test guidelines or any other types of standardized methods are applicable to advanced materials, nanoplastics should also be part of that endeavor, I think. Also, of course, there's a number of complexities because of the nanoplastics being not necessarily homogeneous in the same sense as something that is manufactured to be so. But there are still so many questions to be answered, and I think there's so much that is very, very similar between these fields. So I definitely urged you as a project and, of course, the field to engage maybe in the advanced materials discussions on some level.
Yeah, thanks a lot for the suggestion. Yeah, Amira?
No, yes, I just wanted to add after the considerations from Alberto and Penny, because, for example, in food contact materials, we are also trying to draw the workflow of in vitro BSAs for the risk assessment from some guidelines published from the EFSA in 2019 was the last one, sorry. And it's true that it's about the risk assessment of NIAS, non-intentional added substances, which is also something that concerns a lot the industry of food contact materials, because are substances that also don't we don't know them, and they are also more probably to be in recycling materials, and it's the future also for food contact materials. And it was something that was also a problem, that if I don't know what is going to migrate, what can I use as controls, as positive controls in this case? So normally we work with substances that are considered Kramer-free in the toxicological risk. So now we are also trying to see how we can perform it in micro and nanoplastics. In nanoplastics, as you were asking before, Julia. So it was a very interesting question, because it's something that it's very important to consider when we are doing an assay, also because we are talking to improve the standardization for the industry, for the regulatory. So we have also to consider the blanks, the positive controls, and so on, to be more robust as assay.
Yeah, thanks a lot for your thoughts. Another thing that was discussed, well, it has been also suggested today and also yesterday, is that we should also incorporate or try to include not only experimental NAMs, but other types like grouping strategies, read across and grouping strategies. But my question is, this is general to all of you, what are the parameters? Because as we pointed out at the beginning, if at the moment the biggest problems we are facing is the pollution or contamination, what are the relevant parameters? The plastic that as it is produced, or what is at the end? Should we focus more on the release of substance, bioaccumulation? How could we organize the grouping to be really relevant? And I see that Bjoern, I think you want to answer something.
This is a complex question, and there is no simple answer to it. I mean, you can approach it by different direction. You can think in different use patterns, for example. You could think of exposure patterns. In the end, we will definitely need a grouping approach, especially when it comes to some kind of a chemical regulation like REACH, because otherwise you will not be able to deliver all the data which is necessary to come to a robust hazard assessment, risk assessment, etc. So you need some kind of smart ideas how to group the substances. And Alberto also mentioned that you have the surface, you have the shape, you have, we heard it yesterday, weathered particles, whether natural particles. So it's really a difficult task. And in the end, I think you have to think about a bit what is your final goal. So where are you aiming at? If it's a toxicological effect, then you of course should check what has a certain similarity there, and it could be approached initially by in vitro approaches, by omics approaches, as Penny said, and maybe omics can help you a lot here to check for certain similarities. If it goes for articles or for exposure in the environment, mismanaged waste, I think it's possible to group via uses and to go that route. And I think you should do one without leaving the other out of sight, because we will not solve the issue with only one measure in the end. At least this is my personal assumption.
Yeah, thanks a lot, Bjoern. Penny? You were
Yeah,
you have to answer.
Yeah, I just wanted to say I don't have an answer on how to really fully do it, but I do have sort of suggestions on how to contain the grouping approaches. And so grouping needs to always be hypothesis driven. So it has to have a purpose and a domain of applicability, basically. And if you look at the Commission recommended or JRC developed SSBD framework, you do have these compartments where you can base your grouping on different levels of information. So because you both have the different steps in the SSBD approach, which go through intrinsic hazard assessment, which has been criticized by industry, I know that, and then going to exposure, occupational exposure, environmental exposure, and so on. And those steps are one side of the coin. And then you have also the innovation steps, or stages, actually, because you go through different stages, and the SSBD framework, or the steps in the SSBD, are done for each of those stages in the innovation. So for each and every step, you have a basis of information that you have regardless of whether you're considering safety and sustainability, because you're anyway doing the innovation, and you have structural information, you have functionality information on the materials. So now I'm speaking from the nanomaterial and the manufactured nanomaterial point of view. When it comes to plastic, this becomes different. But so when you go through these steps, you have these different parameters and all this knowledge that is applicable to grouping, and that goes to use scenarios and to life cycle aspects as well. So the further down, and maybe what we as researchers coming from safety and the others coming from sustainability are pushing for, is that a lot of the information is already there for the purpose of the innovation itself, for the product, the end product. Why not use it also for understanding the safety and sustainability in the same go? Of course, it needs data, it needs fair data, and preferably AI to support you in all those decisions. But that's kind of the basis of grouping, that you utilize all this information that you already have, you group as much as possible, and perhaps later in the stages and the steps, you start generating safety data, not for the purpose of risk assessment and not necessarily along the lines of information requirements and regulation, but for the purpose of grouping on a bioactivity similarity level, or maybe it's a functionality aspect. Maybe you're even doing something. Medical development is already doing this. They're already looking at different types of grouping strategies early on, because clinical trials are so extremely expensive. So, it's all a matter of utilizing the data you have as much as possible. And so the grouping scenario really depends on the stage and the data available and the costs that you're prepared to take for generating perhaps a little bit more data beyond what is already there.
Yeah, thanks a lot. I think, Alberto, you wanted to say something?
Yeah, but I saw that Amira was actually before me, so I don't know if you don't have a question, Amira, or...
No, it's because I was also writing some things while the other colleagues were talking. It's also that we are at the beginning, I think, of all of this risk assessment of nanoplastics, and I think it's we have to work hard in this scenario, and also at the beginning. Maybe I sound very pessimistic, but like at the beginning, we have also to consider the specific cases that we have in front, because at the end we are talking about materials that have different manufacturers, and this is very complicated also when we need to trace all the material with all the additives that are added to our material. Also, for example, we can have or we can use the existing regulatories that we have to inspire ourselves to know where we want to go, because for example the ISO 10993, the one with for medical devices, is also one of the most right now used here in the plastic market, but also is one of the most complicated to go. I don't know, Alberto, if you want to correct me, because I think you have more experience than me in this field. But it's true that it's we are talking about the regulation of medical devices that has been published in 2017. We have a field where we find low number of consultants in this field, so it's very complicated also to go further in the in understanding this regulatory, for example. So also I think we can inspire in this we can have the inspiration in the field of micro and nanoplastics, because I think that we are more focused in the human health considerations and environment considerations, and I think that it's a big chance now that we are all together, and we can go further in different projects and different intercomparations and different works that we can go together to know more about this field, because we have to learn about the past, because normally or my consideration is that sometimes we tend to separate the different things, the different fields, when we are analyzing material, in this case nanoplastics. So I think it's important to learn from the past in this sense, because we have to work together, research and development, universities that you have the know-how that is a big know-how, that then from the technological centers and the industries, we have to listen to you, and also we can go from to talk to you about the demand that we can have from industries. And then with all this strength together and all this work together, we can go to regulatory. But I think that we have to work in this sense all together, because at the end you find clients or industry that they don't know how to do with a new material, and then you can find also investigations in university that they end there, and they were very interesting. So now I think it's the moment to focus in the evaluation of micro and nanoplastics and go further with all these roundtables and meetings that PlasticHeal has organized. So I didn't answer anything, but I wanted to.
Thanks a lot, Amira. Alberto?
Yeah, no, just to complement this, I think it's very nice, Amira. I mean, you raised like some very good points. I think one of them is like about very like a quick thing, and like the link between like human health and environment. I think the One Health approach is very important. The two things are connected. But well, my question, the reason why I raised my hand before was fully support and fully agree with Penny about like the use of read across, for example, in the link with SSPD. We have some projects, for example, on SSPD of plastic-related materials that is clear that we need these methods, because we can't test, for example, using experimental methods everything or all the formulations that they have. Because sometimes it's a lot. Just in terms of plastics, for example, as I mentioned before, so we have the plastic polymers, but then we have, different cocktails of additives that we can introduce in the plastic polymer to make them more flexible, more brighter, or with better properties, for example. It's really, really complicated to test absolutely every formulation. So I think all in silico methods, all initial approach, all low-tier methods that we can apply, for example, in the case of SSPD, especially, for example, in the design phase, the early design phase is very, very important. fully support, the use of NAMs, as I mentioned before, so we need it because we need quick methods. We need, methods that that show, like, for example, low cost, to do like a quick screening of the different formulations in an early stage, for example, of the design process. And we have experience with some projects, and and it's interesting, for example, to see how broad this discussion can be, right? Because we talk about, for example, food contact materials, but we also have to talk about other applicabilities, or other applications of these materials. So we have here SSPD projects related with food contact materials, but also with something very maybe different for most of us, but like fishing gears, really like focusing, for example, in the environmental stuff. There's a lot of discussion, for example, on the use of the typical polymers, for example, in fishing gears, and all the discussion about the ghost fishing nets, in the ocean. So we know that at least like people more involved with the environmental studies, that there's a huge issue with the loss of fishing gears in the ocean, because they can remain for many years, and they can trap animals in the ocean. So now the discussions are really moving towards like the use of different and more intelligent, for example, polymers in these fishing gears. So some of the ideas are basically related with the use, for example, of biodegradable materials, or even like materials that you can recycle in a more efficient way, And this is important because, of course, when we talk about biodegradable materials, you are talking about something that you can use for a certain number of years, but then this will degrade. But degrade in what form, in the environment? So you can think about that this probably won't disappear completely. So you can think about that the fishing gear can disappear in the ocean, but can become microplastics or nanoplastics, or can release additives as well. So it's really important to really pay attention to this. So all this discussion and these projects, for example, really focus on using and applying the framework of the JRC to how to really design these new materials to be really efficient and safer, and definitely more sustainable, and for future applications. So the plastic discussion, I think, is very complex, because we need to think about different elements, right? So we don't have only like PP, polypropylene. We don't have only polyethylene. We don't have only polystyrene. We have these polymers, but a mixture of many different molecules, to improve, the performance of these plastics. Just a very last example, but important to mention. A lot of discussion about the use of bioplastics, right, for many different applications. So some of the results, and not all the results, but some of the results that we are obtaining right now, for example, in the different projects, we are seeing that actually, the burden of additives that we need to use, for example, in the bioplastics, sometimes make them worse options than actually the typical plastics, because the number of MEAs that we have that can be released, for example, from these bioplastics, make them in some cases like more toxic than the conventional plastics, because basically to improve the performance of these plastics, and then I can maybe name some of them, like as the villains maybe, but for example, PLA, we are seeing some, bad results actually on the use of PLA, because they can be quite toxic, and sometimes more toxic even than, polyethylene or polypropylene. And then, of course, like linking to the very beginning of the discussion, so the design in the context of the SSPD is really important. Anything that we can use in a very early stage, of methods, in silico methods, or read across, anything like that, I think is key. It's very important to really select the best formulations, for example.
Thanks, Alberto. Soon I will think that you don't need any moderator, because somehow all of you tend to answer the questions that I was planning to ask. Amira, before you are talking, because my next question to all of you was about bioplastic, biocomposites, but I see that there is an interesting question in the chat from Alice Wilson, that please we should clarify if we are referring to bio-based plastic or biodegradable plastics. So I don't know who is the best to answer this question. Maybe Amira, you have a lot of experience. What is the difference if first you can or Alberto, if you can introduce us, what are the differences among these different concepts? And then we focus better the discussion. Thanks, Alice.
Alberto, go ahead if you want.
Yeah, in my example, basically the example that I was talking about biodegradable plastics. But I mean, of course, it's a long discussion about bio-based and biodegradable. But in this case, for example, the applicability in this specific project was biodegradable plastics.
Okay. And coming back to these compounds, also to biocompound, I mean, mixture of plastics, for instance, with other natural fibers, like cellulose or similar. So how do you, because this is coming also connected to the comment of Penny, whether we should consider them advanced material. And I agree that nowadays there is some combinations that include not only plastic material, but mixture of them. So yes. in comparison to assessing plastic, I think in PlasticHeal, Alba, correct me if I'm wrong, we have focused also on polylactic acid, but of course we haven't had time to explore other type of biodegraded plastics or bioplastics. But do you expect, again, do we have differences between what they are even called conventional plastic materials and these new types of composites and combinations, thinking about testing and hazard assessment? Alberto, you can continue.
Yeah, sorry, maybe I'm speaking too much, yeah?
No. As far as nobody is cutting us, we can continue.
That's good, that's great. I think it's a nice discussion, Julia, very in line, for example, with all the discussion that we have concerning, for example, innovative advanced materials. I think this is still like a concept that is not completely defined. Different people have different concepts about what is advanced materials or innovative advanced materials. But I think, for example, taking the information from maybe different forums, and the last one was basically the meeting that we had in Brussels, I think it was last week, of the future potential new partnership in advanced materials, innovative advanced materials. So that will be operated by the IMI, so about like the innovative materials. So I think, for example, of course, if we think about microplastics and nanoplastics in the context that we discuss here, for example, in some potential contaminants, I don't see them as advanced materials, but maybe the plastics can be innovative advanced materials, because if they are designed for a specific application with a very specific function, and they are improved in some way in terms of functionality, for example, they could be considered as innovative advanced materials. If the microplastic or nanoplastics is developed like a microplastic or nanoplastic for a specific functional application, so then they could be advanced materials. But the fact that we have a plastic, this plastic degrade into microplastics and nanoplastics, of course, they result in secondary microplastics and nanoplastics. I don't see them as innovative advanced materials, right? Because they're actually something that come from these materials. But it's very, very important to discuss about the advanced material that could be the plastic per se, because the plastic material can be designed for a specific function, can be improved for better function, for example. And this is relevant because at the end, even degrading, for example, in the environment, we have the release of particles that, of course, will contain, mainly the formulation of this original plastic. So I don't know if I was clear enough, like with the discussion, but well, maybe just more. Yes.
Amira?
No, wait, I wanted to add some things, but I'm going to talk about as you were saying also, Julia, before, the cellulose, for example. It's true that we have had some materials, some bio-based materials. And it's true that, for example, from 2023, there is a guideline published from the EFSA for bio-based materials, for food contact materials, which is, I think, an advanced consideration, because you can also have a workflow of the in vitro bioassays that must be have done. Also, it's very important, for example, that I wanted also to talk before this question of bioplastics, that also mentioned the CUSAR models, because, for example, for micro and nanoplastics in the environment, in soil, water, it's true that they combine a lot in all the projects, CUSAR models and in vitro bioassays, to predict what happens, and also to give feedback to this database, that maybe it's the moment also to enrich this database to know about the material, about the additives, about the different components that are in the polymer matrix. So the CUSAR models are getting force in environment, and I think also in human health, because, for example, it's also considered for some chapters in the standard of the 10993 in medical devices for the skin irritation and skin sensitization, that still uses sometimes animals for the safety evaluation of medical devices. But then talking about the cellulose, it was very interesting, this case that I have in the laboratory, because it was a bio-based material, okay, that has no genotoxicity effect in my cells. But after the migration, I saw that the material was like degrading a bit. Okay, so this is very interesting, because we need also to maybe include the sensorial test, because maybe it can change the flavor of the food that is inside. But also it was very interesting because it was no cellulose, the coating, it was nanocellulose. Cellulose is included in the positive list of compounds that can be used in food contact materials, but nanocellulose is not included. So it's very interesting, because you can learn a lot about all this about all these things, and I think that these cases can also inspire us when we are talking about nano coatings or nano fibers that can be also considered as nanomaterials, or the risk assessment that we are trying to design for micro and nanoplastic can be also then used from all these cases that we can have in the laboratory, because as we were saying, that we have to do much regulatory policy, industry, researchers, technological centers, we also have to do much from all the cases that we have in the laboratory, because it's working when you can also find your muses to go on.
Okay, thanks a lot, Amira. Bjoern, do you have any comment from the industry perspective about this bioplastic, biodegradation, different compounds? There is an active discussion in the chat, I can see, related to that.
Yes, it's impossible to see everything.
Yeah, I'm trying, but it's difficult. Yes, I agree. So Bjoern, if you have any comment about, and after that, I would also like to invite the rest of the audience. If you want to say something, I think Virginia can give you voice and image. So please, if you want to contribute to this discussion, you are welcome. But first, I would like to know, because Bjoern, what I know is that, for instance, BAS is working a lot in biocomposites, including plastics. That's the reason. Yeah.
Exactly. I will try to make it short to leave a little bit room for other comments. And I think these materials can make sense for certain applications, but just to echo Penny a little bit, you have to check in beforehand, safe and sustainable by design, if this is really the best material for your application. It could, if you look only from one dimension on it, if it's biodegradable or not, for example, then you could leave out some aspects which might be important or very important in the end. So you have to look at it holistically, and I think safe and sustainable by design is offering a kind of a framework to do so, because it is taking into account different data. So I think, yes, we can make use of them for certain applications, but possibly not for all applications. It's the most or the best solution.
Yeah, I think that Francisco Ferrer from 9+ in the chat is also, sorry, I cannot read everything, but is also giving similar idea and giving examples where biodegradable polymer makes sense or not. So I guess that, yeah, we are again depending on the use as you have pointed out before. But the problem is that we have been asked to make the risk assessment of micro and nanoplastic in general. So I think that the conclusion we are reaching is that maybe we need more specific topics, or what is the risk assessment, what is the focus for, in order to do it properly. So coming to the NAMs that we have been presented here, now again is a common question to all of you. Could you tell us what you like, what you dislike, or dislike, what is missing, how we could make even better use? Well, Penny, you have marked already some points of how we could make a better use of all the information that we plan to have at the end. But also, if you or the other speakers have any suggestions about the models we have used, for instance, the 3D models or simple models, Hans has pointed out an interesting point. It was, as he said, in previous projects with nanomaterials. Having in mind the huge variety of plastic, does it have any sense to create some kind of pre-screening with simple models to identify what could be, let's say, the most dangerous plastic, for instance, based on oxidative stress, and then focus on them, somehow make some prioritization, because neither we can test everything. So could we have this kind of prioritization system for the hazard assessment or something like this? Penny?
I was going to say something in relation to all the NAMs and the really amazing systems that you use, but I will address what you're asking now. So I was happy to see Hans presenting AOPs. And I actually wanted to, Sabina was talking yesterday also, and she's been working on some AOPs, I think also in relation to nanoplastics, actually. And since, so the benefit of AOPs is that it supports you to structure your data in a good way. There's, of course, on one hand, there's the need to verify the data and make it really truly available. That's a huge endeavor. And since you're at the end of the project, I'm not sure how much time you will have to truly engage on that. But I would urge you to at least have some sort of, so that maybe a follow-up project can access the data on some level. And you might look at, for example, Park, who is developing the project Park, the big EU project, who is working on fair data. But if you look at AOPs, that allows you to really structure, and it also actually builds or supports you to build integrated testing strategies, or even integrated approaches for testing and assessment even. So if you during the last months of your project, if you have the opportunity to map your assays to AOPs, I think that might be something very valuable. And especially if you've developed new NAMs, they will be broad, they will provide bigger or broader impact if you have them connected and anchored to AOPs. Now, there's, of course, many ways of doing that. You can do it on computational ways, allowing people to get access without even knowing that they're there. But, of course, also publishing just a paper demonstrating the connection to AOPs is valuable for follow-up projects. So, yeah, just an idea.
Thanks a lot. Yeah. Alberto?
As again, fully supporting, for example, the use of NAMs, we need this. We need this for example, for the first initial screen, for example, of the materials. We can't test absolutely everything, So I think it's really, really important. We, in Geico, for example, we mostly use, for example, in vitro methods, to, support, for example, the selection of the screen, for example, the different materials. And I think they are quite valid, and we show like very clear, results in the application, not only, for example, with to assess, for example, acute responses, but also chronic, chronic we used to call, for example, in vitro world, but our chronic is not really the chronic of the in vivo people, so it's a critical. So we need to really maybe to rephrase this using more like short term and long term assessments. So normally we go like up to 28 days or a month of exposure in vitro, but this is very short period for the in vivo people. I think this definitely is like as a great tool, for example, to use, so great potential not only for the human health assessments, but also for the environmental health assessment. We used to call environmental health assessment as well. I think it's just like important, for example, to really understand like the application of everything, because we know based on experiences that some things can be really, really tricky and can be complicated, in the design of your experiments at the beginning, So coming back to this first point that we mentioned like at the beginning, some considerations, I think they really need to be more stressed and they really need to be more discussed, for example, for the experiments, right? So we spoke, for example, initially about the behavior of the materials in the different exposure medium. We need to, maybe we need more information about the characterization of these microplastics and nanoplastics in the different medium. Do they release the additives, for example, in the medium that can cause like a parallel effect? So this is one of the things. Another thing that we need to discuss is also that microplastics and nanoplastics, they are everywhere, right? So do we have really like a standard and really well-controlled environment in our laboratories, for example, to avoid a secondary exposure, to microplastics? Because we have our experimental systems, we add our microplastics and nanoplastics, but do we have, are we sure that some microplastics and nanoplastics are not coming from the environment or even from the, systems that we use? For in vitro, most of us, of course, we use plastic plates, so are we sure that, for example, some of these microplastics or nanoplastics are not coming from the microplates, or even like, for example, from the bottles of the cell culture medium? So there are so many, small things that we need to ensure that we will have a response at the end in the cells, So I'm not invalidating, for example, the NAMs, but it's totally the opposite. I think is they work, and they are, of course, they are very useful for this, but we need to be sure that everything that we do, for example, in terms of, really, setting this experimental system is really well done at the beginning, because otherwise, the response that we can get at the end can be very different. Julia, like some experiences we have with previous projects, that is not simply, for example, like suspending your microplastic or nanoplastic in a cell culture medium. We need dosimetry. We need to know how much of this material is actually in contact with the cells, because we will definitely get a response in the cells at the end, but maybe this response is related with 10% of your material or 100% of the material that you put in the system. So I think all these, quality control things, they need to be really assessed at the beginning, in order to have a very good experiment and in order to really ensure the quality of your results, because results you have at the end. But the thing is that, is this really precise, in terms of experiments? So short answer, I really believe in NAMs, but I really think that we need to put more attention, for example, on the design phase of the experiments. That's we have a lot of experience, for example, based on the nanomaterials from the nanotox world, for example. Some of them, they are basically very similar here, but not all of them.
Yeah, thanks, Alberto. Bjoern, what do you think about our NAMs? Alba has also launched a question in the chat about that. What could be the first essay to consider in a prioritization system, or how do you do it? Yesterday we were talking about the huge amount of plastic. So if we have to start, how could we like select who are the bad guys and not so bad guys, let's say?
Yeah, it's a difficult question. I'm also optimistic that we will make use of NUMS, of modeling, of combined approaches. It will be not so much one essay who answers one hypothesis, but it will be a set of, or kind of a toolbox to use. And I mean, as an example, we internally, we sometimes use omics, metabolomics in this way to check for similarity in the patterns. So does a certain innovative substance has a certain pattern? And then we see, okay, here might be an effect on the liver or on something else. And then we can do a targeted testing on that. And I mean, coming from the structure, you can also have some primary alert, and that could help you steer a little bit the direction which you want to aim. But in the end, yes, it will be very complex. And from my understanding, it will also be much more complex than, let's say, the current REACH testing regime, which we have, which is very much hierarchically ordered. It will be much more complex in very diverse directions. But it's, I think it's the only way to go, because we will not be able to fulfill all the data requirements and needs, not only for regulatory purpose, but also for safe and sustainable by design, with the old-fashioned approaches based on vertebrate animals only.
Yeah, thanks a lot. Yeah, well, in this second day, as you have seen, we have focused on the NUMS, but we have also showed you some results from the biomonitoring studies that we have performed in PlasticHeal. We have also analyzed the effects in these populations that you showed today, but we are still compiling all the data, and these results will come later. But at the moment, it was just to give you an idea what are the levels that we are finding in these populations. So as Alba has also launched a question about, what do you think about the results of our studies? What we have seen about the levels of micro and nanoplastics in the external, but also in the human samples? Because, for instance, from Irina's talk, one of the conclusions was that the fact of living in an area has higher impact in the level of micro and nano or nanoplastics in people that know the fact that they were working or not in the factory. So what do you think about this, or how, and this is also connected with what Alberto talked, that the problem we have with micro and nanoplastic came not only from our equipment. In the case of human biomonitoring studies, what we are eating, drinking can be more relevant what we are working in. But yeah, Alberto, please.
Yeah, no, it's a very good point, Julia. I think it's important to remember that, for example, exposure to microplastics and nanoplastics is not a recent thing. I mean, we've been living with, plastics for, decades and, many decades. And so this is, we are constantly exposed to this. It's a very, very, interesting story about the biomonitoring studies that you guys have in PlasticHeal, where we have similar experiences, even like with the ecotox side, So, for example, we collect animals to perform experiments in the lab, exposing these animals to microplastics and nanoplastics. And then we see that the actual level of microplastics and nanoplastics that they already contain, in the different tissues, sometimes they are high, because these animals, they are not like living in pristine environments. They are exposed to microplastics. Even the negative controls, they already have, the microplastics and nanoplastics in the systems. So we need to think that these animals, they don't come like with that unexposed condition, right? So they have as a background the microplastics and nanoplastics. They are probably exposed to the additives as well of these microplastics and nanoplastics. And they are actually, in your experimental system, maybe with a higher level of these materials. And I imagine, for example, in the biomonitoring is more or less a similar thing. So these people, they have a background level of exposure to these plastics, right? the fact that they are exposed in these, work environments to more plastics, for example, of course, probably the effects are higher. But I mean, we have, a base ground level, for example, of responses that are already there. So,
Yeah, thanks a lot. The other speakers, do you have any thought about this, the first human biomonitoring results we have shown you?
Yeah, but maybe one point, as a scientist, I would be interested to see if there are already epidemiological data. I mean, these people shown in this occupational environment, they are probably exposed not since yesterday to a higher level of polymers in whatever state. And it would be interesting to see if there's a significant increase in certain occupational diseases around that, because then you would have a direct link and a causal link to really the disease. That could also help in the end thinking about regulations now setting threshold levels, because as you said, Alberto, we're not the exposure that we as humans have, most of us, is since decades. And if you have special cases with a significantly elevated exposure level, you might have some real life findings there, which you can then go back to the lab and correlate to your findings and then check what is really the hazard combined to the exposure, what is my risk? Because this is in the end what regulators are about and what they want to know and what they are building their decisions upon.
Yeah, I think good point here, because I think this is another thing that was actually, I think it was present in some of the presentations, but also, for example, the possibility of these particles and to act as Trojan horse, carriers of contaminants. I mean, different contaminants. So we can think about, for example, pathogens, or we can think about, for example, even like organic contaminants. I think PlasticHeal, you guys do some work on Trojan horse effects as well, right? I'm not totally sure. But in our case, for example, in plastics fate, we do a study, we do evaluate these effects as well. And we see that the plastics, in many cases, for example, they can carry different microorganisms, for example. We see, that's in many microplastics and nanoplastics made of different polymers. So especially, for example, polyethylene in PET, we see the formation of a biofilm of pathogens, for example, on the top of this plastics, and then clear effects linked with these pathogens in the cells. And then an interesting event as well, of course, is that, I mean, some of the experiments we run, for example, exposing the models to the plastics with the biofilm of the pathogens, or even to the contaminants, for example, PAHs, like polycyclic aromatic hydrocarbons, for example. But we see that the effects exposing the models to the plastics with the biofilm or these contaminants on the surface of the plastics, they are higher than, in many cases, just exposure to the pathogens or just to the contaminants, which means that in some cases, for example, the plastics can accumulate higher quantities of these contaminants and higher quantities of these pathogens, provoking, for example, inflammatory effects, or even effects more related, for example, with the presence of these organic contaminants, in some cases, for example, genotoxicity. So this is, I think, is very, very important and very, important to discuss, because it's not only the microplastics, but it's everything like related with this in the environment, right? Allergies, for example, the increase of allergies. I don't know if you are evaluating this as well, Julia, in PlasticHeal, for example, in terms of increase of allergies, or a possible relationship of the exposure of these workers with the increase of allergies, for example, or something like this?
At least we are assessing the increase of some cytokines. So we will assess the inflammatory response and genotoxicity response also in these workers, and compare with the control population. We'll see if there is any difference or not, of course. Yeah.
In our case, for example, in plastics fate, we do see, for example, the attachments of LPS, LCA to the surface of the plastic particles. we do see the formation of the biofilm of bacteria, for example, or fungal particles, and anything like that. So we can see that actually these particles, they can accumulate these different elements. Then we can see effects like, for example, allergy or inflammation or other things. So I think it's a very good thing that, for example, the CASP projects are really like bringing, you know, to the discussion, not only the CASP projects, of course, like many other projects on microplastics and nanoplastics.
Yeah, you are right. And in fact, what we are trying, at least at the CASP level, because of course the human biomonitoring studies are also challenged by the size of the populations of the group that we can assess. So what we are trying is to combine effort from the different projects. This is something we will discuss in the CASP meeting, in the final CASP meeting, trying to harmonize as much as possible all the knowledge that we can, so that we can provide at least some indication about the human data information that we have. Penny, I saw you like wanted to say something, or okay.
No,
Okay,
I keep thinking around this, and you know, I see parallels also with PFAS, which of course, you know, we didn't know what. And now it's all about endocrine disruption, but before that it was just a row of confusing endpoints that didn't really fit into the regulatory frameworks. And so maybe we have something similar here, that it's also difficult to assess what the combined devastation is, rather than the single adverse outcomes that we see for certain types of, like there's so many different mechanisms and combinations of bacteria bringing plastic into the body or vice versa. And we've seen that was also in a hypothesis regarding asbestos and tobacco smoke, for example, that asbestos brought carcinogens from the tobacco closer to the vulnerable places in the body and to cells. So, you know, we don't really know. And the combined effect of all these different types of mechanisms is hard to handle and calls for a precautionary principle, but very difficult to set one, of course.
Yeah, that's the reason also in PlasticHeal we are trying to combine the different approaches. For instance, Alba was telling today that the omics will be also done to some extent, some biomarkers from the human samples. So we will compare what you say, this complex exposure that many things can be involved, with the same biomarkers with some simple in vitro assays. So we can see if really there is something reproducible, if we are detecting different things, so what could be. We will not have the answer, but at least we have some clues what are the next steps to take. Yeah. Okay, I think we are reaching the end. So my last question is if you have any other comment, question. The audience has been quite silent today. Well, the chat has been very active. I couldn't follow, but if anyone in the audience wants to say something, or if you have any question you would like to raise or comment to do, these are the last words.
Yes.
Sorry. Yeah, Amira?
No, the chat was incredible. It seems tender, Julia. But the thing is that I wanted also to have the chance to talk now that we are all together here from PlasticHeal, and also the audience that I'm very grateful to have shared this morning and also yesterday morning with all of you, and all these questions about the nanoplastics, bioplastics, and so on. But I also wanted to have the chance to consider the second part of this PlasticHeal for the future, as we were talking about, and also Alberto mentioned many times about the environment and all this strategy of zero pollution in the cluster 6 of European projects. That it's a I read a topic that can fix very well with continue of the PlasticHeal. And it's very important also, as we are here in Valencia, and we are working hard in the management of waste after Redana, and how the concern of society also is focused in the micro nanoplastics that come from all this waste, the emergent contaminants that are going to finish in our sea and in our albufera here in Valencia, and I think it's going to be a inflection moment to consider, and also because the consumer and society is getting more involved in what environment means for us, for human safety and for human health. So I think it's also important to consider these advances that you have done with PlasticHeal, with human health, which are incredible advances, and to go on and focus also in our home, that it's our planet, and design all this battery of in vitro or NAMS methodologies to determine the risk assessment and the safe and sustainable by design materials for the consumers and for environment.
Thanks a lot, Amira. Alberto, you can continue.
Just a very last comment. I think it's a very interesting discussion, because I think we have to move, in my opinion, from the point of just identifying the hazard of the microplastics and nanoplastics. So maybe it's, move to another discussion that we actually, many forums, we are actually already doing, but can we support the plastic industry, to develop safer and more sustainable plastics for future applications, so more, better materials for the application. So this is for me is like important. Plastics exist, the application of plastics are here, so we are not here only to point out saying that these microplastics and nanoplastics are bad. I think we have the potential to maybe using our techniques, using our NAMs, for example, to support the industry to develop better, materials.
Yeah, I fully agree on that. Bjoern, any last comments? Oh.
I think I'm fine. My previous comment was my last one, I think.
Okay, then thanks to all of you for this nice discussion. Sorry if the questions were a bit tricky, but I think that you have handled really well. And now I will like to give back the floor to Alba for the final words.
Okay, thank you very much. Well, I'm going to close the meeting just by saying that it's been amazing to discuss with all of you these 2 days. I think we are getting with us a lot of insight and things to reflect on in the coming months for PlasticHeal. And I hope that you will follow up on our developments, and you will see how we will incorporate as much as possible all this discussion into our next steps. And thanks also for the PlasticHeal Consortium. I've said many things that you are doing a great job, and it's been reflected today and yesterday with the different presentations. So thanks to everybody. And Virginia, I don't know if you want to remark something regarding the coming of the materials after the meeting that I believe we will share with participants, just as for them to know.
Yes, basically is what I have already said in chat, that remember that we will be sharing all the information that you have generated with your participation, really active participation, for which we thank you a lot. We are very happy with the results and of the discussion, and we hope that in the coming days we will be sharing all of this with you. I will let the new toy open for for further discussions. I mean, the whiteboard will remain open so that you can, if you forgot to mention something, or if you still want to comment something else, I would like to let you know that the members of the PlasticHeal Consortium will receive the whiteboard and will be able to answer any further question that you may have. So thank you, and see you in February in the CAS meeting as well.
Thank you.
Thanks a
Bye,
lot.
everybody.
Thank you. Thanks.
Thank you.
Bye-bye.
Thank you. Bye.
Bye bye.